Hello everyone. Am I back? Can you see me? Can you hear me? Can we continue? Let everyone join. I will start in few seconds. Okay? Let everyone join. So everything is clearly visible, audible. Mahima, Farhan, everything is good to go. You can see me. You can hear me. The slides are also visible. Can we continue? We will start in few seconds. Let students join. Otherwise, some can miss the initial portion now. Just few seconds. Okay, okay. Thank you. Thank you for responding. Thank you for your concern. Thank you. So, can we continue? So, let's start with the replication of the viruses. So, we have started virology, right? So, we have started virology. In the virology, we have seen the classification of the viruses. So, we have 6 families of the DNA viruses. We have 15 families of the RNA viruses. We have seen many things in them, which is single-stranded and double-stranded in them, which is smallest and largest in them, which is segmented in them, which is enveloped and naked in them. We have seen all these important points. After that, we are coming on the next thing, that is replication of the virus. Do you know that viruses replicate only in living cell? Please learn this line. Viruses replicate inside living cell. Viruses cannot replicate outside the living cell. So can I grow the viruses on the agar plate? This is the agar plate. Can I grow the virus on the agar plate? What does the agar plate contain? Agar plate for the bacteria, for the culture media, for the bacteria, it contains protein, carbs, lipids, fats, everything, but it don't contain any living cell. So agar plates of culture media are non-living media. So viruses cannot be grown on that. So if you put bacteria on that, bacterium inoculation, you will get the colonies. But if you put virus on that, virus will not divide. Virus will not divide on agar plate. This is the reason culture media are non-living. uh, they don't have living cells in them, right? Now let me come on the replication. You must understand the replication of the virus. Can you see this yellow color cell? This cell is a host cell. Let's assume the host cell is human. It can be animal also, it can be human also. For example, this is a human cell. So see the 6 steps. How does the virus is doing the replication? The first step, the virus is attaching. This is known as adsorption, attachment with the help of the receptors. So peculiar receptors are there. It is not the thing any virus can bind with any cell. No. For example, HIV. For example, HIV is a virus. This HIV virus bind only with CD4 receptor. CD4 receptor present on the T lymphocyte. That's why the target cell is CD4 T lymphocyte. It cannot bind with any other cell. In the similar way, influenza virus. Influenza virus is a virus that bind with the glycoprotein present in the lung cell. The glycoprotein present in the lung cell. Otherwise, it cannot bind with other cells. So that's why influenza virus involves the lungs, not other organ. So every virus have a peculiar receptor present on the host cell with which it is joining, it is attaching. Got my point? So this is attachment. This is attachment, adsorption. After that, virus is penetrating. Virus form a vesicle. With the help of the vesicle, it penetrates. It penetrates inside the host cell. After that, the third step is uncoating. In uncoating, the capsid and nucleic acid separate. Can you see this is nucleic acid? Can you see this is capsid? So capsid and nucleic acid both are separating. And this is known as uncoating. The fourth step is must, very important step. It is biosynthesis. So multiple copies of the nucleic acid are formed and multiple copies of the capsid are formed. This is known as biosynthesis or replication. You know all the materials required, the enzyme, the metabolite, the protein, whatever is required for making multiple copies. All the material is taken from the host cell. कहने का मतलब सामान सब हमारा और जो प्रोडक्ट बन रहा है वो वायरस का. So all the material which is required is of host cell, but the material which is produced, the product which is produced, that is of the host cell. Host cell की प्रोजेनी बनाने of the virus है ना. So virus की प्रोजेनी बनाने के लिए कहने का मतलब this cell is hijacked. You can see this cell is hijacked. तो इसके अंदर जितना भी सामान यूटिलाइज हो रहा है वो होस्ट सेल का है, लेकिन जो प्रोडक्ट बन रहा है वो वायरस का है. Got it? Got it? So that is biosynthesis. The fifth one is the maturation. Maturation also known as assembly. Maturation, also known as assembly, in which one capsid bind with one nucleic acid. One capsid bind with one nucleic acid, like this, assembly. And finally, the host cell is ruptured and the daughter cells released out. The host cell is ruptured and the daughter cells are released out. The last step is the release. The last step is the release. So these are the 6 steps. Now you can imagine what are the 6 steps. What are the 6 steps of the virus application? First is attachment. Imagine the diagram. Attachment, also known as adsorption. Okay, specific receptors are required. Number 1. After that, penetration. So forming a vesicle, it is penetrating inside. है ना? It is penetration. Okay, penetration. Okay, the second is the penetration. After that, it is uncoating. What do you mean by uncoating? The capsid and the nucleic acid separate. This is nucleic acid, this is capsid. They separate from each other. That is the third step, uncoating. After that, the most important step is biosynthesis, in which multiple copies of the daughter cells, daughter virions are formed. These are known as daughter virion. Daughter virion are the daughter cells from one cell. The parent cell, they are formed. So all the material utilized is of host cell. By saman hamara mal tumhara. है ना? That is the point. So it is hijacked like that. The fifth is the assembly. Assembly, also known as maturation. And finally, the host cell is ruptured. Mind my words. The host cell is ruptured and the multiple daughter virions are released out. So what is the end result? End result is the rupture of the host cell. Since the host cell is ruptured, that's why viruses cause diseases to us. Now imagine this yellow cell. Host cell is a lung cell. Person will have pneumonia. This host cell is the intestinal cell. The person have a diarrhea. So because that particular organ cell is ruptured, that's why it is causing the disease. How many of you got it? How many of you got it? Huh? You got it? Can we proceed? So this is the 6 steps. Now please understand one point here. In the 6th step, let me erase. I want to tell you a point here. Can you see the entry of the virus? And can you see the exit of the virus? Can you see here? The virus is entering. This is entering. The virus is entering here. And can you see this is the exit? The daughter virions are exiting here. Hana? So you can see a virus visibly here. You can see a virus visibly here. But during this period, the virus become invisible. The virus not visible inside the cell. It is known as eclipse phase. What it is known as? Eclipse phase. You know what is the meaning of the eclipse? Hindi mein kehte hain grahan lagna. Have you heard about the moon eclipse, sun eclipse, Surya Grahan, Chandra Grahan? At that period, the sun and moon are not visible. That is the eclipse. In the same way, this is virus eclipse. It is not sun and moon. It is virus eclipse. The virus become invisible during this time. This is known as eclipse phase. That can vary from virus to virus. There is no fixed duration. What is the duration of the eclipse phase? But this is known as eclipse phase that you have to learn. Okay, Ji, can we go ahead? Okay. Assembly is one capsule binds with. Swati, the first thing, it's not a capsule, it's a capsid. Hana, capsule is in bacteria. You are right. Technically, you can say capsule, but better you say it's capsid. Hana. So yes, one nucleic acid bind with one capsid. So in the virus, I guess you have attended the initial portion. In the virus, there is a nucleic acid surrounded by the capsid. This is the structure of a virus. So one nucleic acid bind with one capsid. That is the assembly. Got it? Can we go ahead? Hana. So replication is done. Now, cultivation of the virus. What do you mean by cultivation? I want to increase the quantity. See, I'm having a specimen with me. Imagine this is my specimen. Now this specimen contains the virus. I'm assuming it contains virus. My specimen can be anything. It can be saliva. It can be blood. It can be urine. It can be stool. It can be CSF. It can be anything. I'm assuming it contains. The virus. I want to increase the quantity. Imagine if it is not a virus, it is a bacteria. What should I do? You will say, ma'am, it's very easy. Take a culture plate, culture media. Take a culture media, inoculate. The bacteria will get the nutrition on the culture media. It get all the nutrition. It will multiply and form multiple colonies. So it was very easy if it was a bacteria. But viruses do not grow on culture media. Viruses do not grow on culture media because the culture media is non-living. If I try to grow virus on a culture media, this is a virus, not bacteria. Viruses will not grow. No, they do not grow. They do not form the multiple colonies like the bacteria. No. So how to increase the quantity of the virus? I want to increase the quantity. Yes, PCR is definitely one method, Annie. You are absolutely right. But number one, it is very costly. It is not very affordable. It is very costly. And a PCR, it is a molecular method. It is PCR by which we can increase the quantity. I taught you in general, in genetics. But tell me the various ways by which we can cultivate a virus apart from PCR. So what you people are asking me to repeat? You You people are asking me to repeat the eclipse phase. The duration from the entry to the exit. Virus is entering the host cell at this point, and virus is exiting from this point. The duration is known as eclipse phase. This duration. It can be in hours, it can be in days for various viruses. There is no fixed duration. It can be few hours, it can be few days also. Some viruses replicate fast, some viruses replicate slow. The duration is known as eclipse phase, during which virus become invisible. गायब हो गया host cell के अंदर. पूरा it got disassembled ना. Everything is separated. You cannot see the complete virus inside the host cell. Got it, Bilal? Got it, Shivam? Hmm? Got it? Eclipse phase. You people were asking now. Anything else? Can I proceed? You were asking eclipse phase only now. So this is eclipse phase. Anyways. So coming on the cultivation of the viruses. We know viruses don't cultivate on culture media like the bacteria because the culture media do not contain living cell. Viruses require living cell to grow. है ना? So कैसे grow करेंगे virus? How to grow the virus? PCR is a method, I agree. But apart from that, yes, Sandesh, absolutely right. There are three ways to replicate, to cultivate the virus. Please understand the three ways to cultivate the virus. Number one is animal inoculation. Usually we use mice. So take the specimen of your patient. This is the specimen of my patient. I'm taking it in a syringe. It can be blood, urine, whatever. My specimen of my patient. I'm taking my specimen in a syringe. Can you see? And I'm injecting inside the mice. Bechara mice. And I'm injecting in a mice. I can inject intracerebral, see the brain. I can inject subcutaneous, intraperitoneal, intranasal, and I will notice for the symptoms. If mice have the same symptoms like the patient is having from which we have collected the specimen, or the mice died, the diagnosis is confirmed. It is a healthy mice. हमने एक healthy mice लिया, उसके अंदर हमारे patient का sample डाल दिया. अब दो चीजें हैं, या तो mice will remain healthy, so this sample is negative, है ना? Or else the mice will have the same symptoms or the mice will die of the disease. So the diagnosis will be confirmed it is having the viruses. Got my point? है ना? So that is the animal inoculation. Here we use the mice, especially suckling mice. Mice नहीं, mice का बच्चा. Suckling mice is very susceptible for the virus. We get the results very quick. Suckling mice. So we use suckling mice. It's a PYQ. Usually for animal inoculation, we use mice. We are sacrificing the mice. Got it? Now the second is, this is very tedious to do. So we don't perform it. Theoretically it's good, but practically we don't perform it. Okay? The next is egg. Do you eat egg? The same egg. Hen's egg. So second is egg. The hen's egg. Murgi ka anda. The hen's egg. Hana, you can see this is hen's egg. So in the hen's egg, you have to understand something. Can you see in the hen's egg, what is the center? Center contain embryo. The center contain embryo. In the hen's egg, the center contain the embryo. Okay, this is the egg. This is the outer. Hana? This is the shell of the egg. And the center contains the embryo. Now there are four layers. Please understand the sequence. The outermost layer is known as CAM. Chorioallantoic membrane. Say the full form. Chorioallantoic membrane. CAM. Please learn. Okay? Just below this, the second membrane. There are four membranes. The second one. Just remove the word choreo. Only allantoic membrane. So first chorioallantoic membrane, then allantoic membrane. The second membrane. The third is amniotic cavity. And the last one you can learn here, we have the yolk sac. And after that we have the embryo. Say yes. So basically all the four layers, this and this, they contain living cell inside that. All the layers contain the living cell inside. All layers contain the living cell inside them. Obviously they contain the living cell. So basically you take your specimen and inject in one of the layer, either here or here or here or here. So you do an injection. You take an egg. Imagine you are taking an egg and you are taking an injection. You are taking the patient samples in the syringe, with a very thin needle, very thin and long needle, and inject it from the shell. Don't hatch it. Shell में छोटा सा hole करके just inject it. Outer, first one, second one, third one, fourth one. So deeper you go and you just inject in any of the four. Got my point? And come out and let it incubate for the next 48 hours. Next 48 hours, incubate, give adequate temperature. So if the sample contain the virus, the virus will grow. virus will go inside the living cell and virus will grow. है ना? Virus will grow or not grow? The virus will grow. So just suppose you are injecting 10 viruses here. है ना? 10 virus. So all 10 will go in one cell. There is a rule, one virus enter in one cell. One virus enter in one host cell. One host cell at a time have one virus only. For replication, I have told you the 6 step now. What is the last step? The first step is attachment, then penetration, then replication. The last step is rupture. So the 10 cells will be ruptured. If you are injecting 10 viruses, 10 cells will be ruptured. After that, you hatch the egg. 48 hours ke baad, hatch the egg and fletch the membrane out. Take the membrane out. The membrane will look like this. This is not egg. This is the membrane we have taken out of the egg. We have hatched the egg. हिंदी में कहूँ तो अंडे को फोड़ दिया, hatch कर दिया, उसके अंदर की membrane खींच के निकाली, और membrane पे I am looking for the lesions. ये छोटे-छोटे lesions दिख रहे हैं? Can you see the lesions on the membrane? These are the ruptured cells. These are known as POC. P-O-C-K, POC. It is not P-O-X. It is P-O-C-K. What is the pronunciation? POC. So you can count the number of the POC. Here, how many I can see? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, whatever. For example, I can see 12. Means the specimen contains 12 virus. So one POC represents one ruptured cell. One ruptured cell. One POC is equal to one virus. So number of POCs is equal to number of virus. Say yes, people say yes. This is the egg inoculation. समझ में आया क्या? समझ में आया? So these are the 4 membranes. Different viruses are injected in different membranes. Don't learn. Don't learn. Just know the name of the 4 membranes. Chorioallantoic membrane. Just below that, remove the word choreo. It's allantoic membrane. है ना? Just below that, the third one, amniotic sac. और उसके नीचे, it's yolk sac. So there are 4 membranes. Can you see the egg? Appreciate the embryo at the center. Please mind my words. I'm not injecting an embryo. No, 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 no. No, not an embryo. I'm injecting either here or here, or here or here. So in the membranes, I'm injecting one of the 4 membranes. I'm inoculating the egg for 48 hours. After that, I'm hatching it. Hatch the egg. अंडे को फोड़ दो. Take the membrane out. In the membrane, if the sample is positive, you will get some lesion. The lesions are known as POC. Why these lesions are formed? Why? ऐसे छोटे-छोटे ना white color के plague जैसे दिखेंगे. Why they are formed? So basically these are the ruptured cells. Do you know the last step? Ultimately the host cell is ruptured. The last step is release. The host cell is ruptured. Whenever the host cell is ruptured, at that place this lesion is formed. So basically the lesion is a ruptured cell, and one virus will rupture one cell. It is a rule. So point is that one POC is equal to one virus. It is a rule. Learn the golden rule. So can I say it's a quantitative method? Yes, it's a quantitative method. By counting the number of the POCs, you can comment on the number of viruses in your sample. For example, I have injected 0.1 ml inside the egg. So 0.1 ml contain these viruses. So total I'm having 10 ml. So 10 ml में कितना? है ना? 0.1 ml have these many viruses. Just suppose in 0.1 ml I got 9 POCs. So 10 ml will have how many POCs? I can do the calculations easily. It's a semi-quantitative method. Mark the word each POC, single virus. One POC, one virus. Highlight it, mark it. It's a quantitative essay. It's a quantitative essay. Say yes, people, say yes. So animal inoculation we have done in the mice. Say yes. Egg inoculation we have done in the 4 membranes. One of the 4 membranes, not all 4 together. One of the 4 membranes, and we are counting the POCs. We are counting the POCs, the number of the POCs. Say yes. The third and the most common way to inoculate the virus, is the tissue culture. What you are asking, Sandesh, ma'am, is this to determine the number of the virus? Yes, Sandesh, absolutely right. Sandesh, absolutely right. To be specific, इसको और ज़्यादा correct करती हूँ. One POC is equal to one virus. I told you one virus. Better to be say one live virus, because only live virus will cause the rupture. If the specimen contains dead viruses also, that cannot be counted. So with the help of the POC, I can count the live virus, not the dead or killed virus. Dead or killed virus cannot cause the rupture. Okay, got it? Okay, like we can count RBC. Yes, definitely. So you can tell कि आपके sample में, this is my sample. It contains how many live viruses? How many live viruses? You can do the counting by inoculating it inside the egg. So basically take an egg, this is the membrane in the egg, inject your specimen in the membrane, leave it for 48 hours. After that, hatch the egg, take the membrane out, And just imagine this is the membrane I am taking out of this, है ना? And count the number of the pogs in that. 1, 2, 3, 4, 5. So whatever is the number of pogs that me are present here, है ना? So in this way you can count. Okay, Ji, can we go ahead? The third and the most common way by which we do the cultivation is tissue culture. Or the most common way, the third way, that is tissue culture. Tissue culture also known as cell culture. है ना? Listen, listen, listen. So basically what we are doing, take an animal. Any animal. ठीक है? Take any animal. Kill the animal. No. Sacrifice the animal. Better to say. है ना? So we are killing, sacrificing the animal. Take the organs out. Take the organs out. Cut the organs into multiple pieces. Cut it with a scissor into multiple pieces. Now treat it with the enzyme. है ना? Treat it with the enzyme. Which enzyme? You can use trypsin. I know trypsin is there. Various enzymes that will separate the cells apart. All the cells are separated. All the cells are far away. है ना? They are separated. Now take a petri dish. You can take a petri dish and take a monolayer of the cell. Monolayer. Highlight the word monolayer of the cell. This is known as tissue culture, cell culture. I know it's look like agar plate only, but this one is cell culture. It contains monolayer of the cell. Say yes, it contains monolayer of the cell. है ना? It is of three types. The cell cultures are of three types: primary, diploid, and continuous. So here we have the cell now. This is my petri dish, and this is the monolayer of the cell in that. Monolayer of the cell in that, which are obtained from some animal. By cutting some animal, we have obtained that. Now, have you heard about the word senescence? Senescence, I don't know the spelling. Senescence. Have you heard this word? Senescence means after a fixed number of cell division, the cell do not divide. And it permanently becomes silent. That is senescence. पैथो में पढ़ा होगा ना? Aging हो जाता है. So after some time, cell do not divide. So what is the life of this cell culture? So you will see, ma'am, it is having a monolayer of the cell. Maybe after 5 division or 10 cell division, they will not divide. They will not divide and all the cell will die. So what is the life? Life is very less. Life is very less. So we make the cell culture. There are three type of cell culture. Let me draw the three type of cell culture in front of you. Okay. This also have the monolayer. This also have the monolayer, and this also have the monolayer. All the three have the monolayer. Here we are adding nothing. It's only monolayer. So after 5 to 10 division, it will stop dividing. This one is life is very less. This one is primary. This one is primary. And this type is known as primary. But we want to increase the life. So we are adding additional growth factors. Additional growth factors. है ना? यहाँ पे additional growth factors. यहाँ we are adding nothing. Here we are adding additional growth factors. So instead of 5 times, it will divide 50 times. So lifespan is increased, है ना? And these one are known as diploid. These one are known as diploid. लेकिन end तो इनका भी होगा, कभी न कभी होगा. So these are ended very soon. These are ended after some time. After some time, not very soon, but after some time. So the best to use these cells. These cells are cancer cells. Use these cells as cancer cell. You know one property of a cancer cell. Have you read neoplasia and pathology? Have you heard about the word immortal? Cancer cells are immortal. सुना है? हिंदी में कहूँ तो cancer cell अमर होते हैं. अमर. You know who is अमर? The person who never dies. The person who never dies. है ना? No one is immortal in this world except the God. है ना? So no one is immortal. Everyone who is born has to die. ठीक है? This is not a spiritual satsang. So come back. ठीक है? So yes, never dies. Cancer cells never dies. So so one of the scientists said that why don't you take cancer cell? If we are taking cancer cell, it will become indefinite. It will become indefinite. Here only 5 cell division, here 50 cell division, here indefinite. It is indefinite. It will never die. And this is known as continuous. So we have 3 types. What are the 3 types, please? What are the 3 types of the cell culture? Please understand. Say, ma'am, primary, we are adding nothing. After 5 division, it will die. In diploid, we are adding the growth factor. So after 50 division, it will die. But in continuous, we are taking cancer cell. They are indefinite. They will never die. How many of you got it? How many of you got it? How many people are alive, I mean, awaken with me? समझ में आई क्या बात? हाँ? कुछ नहीं डालोगे तो 5 बार डिवाइड करके सेल मर जाएगा. थोड़ा ग्रोथ फैक्टर डाल दो, थोड़ा खाना पीना दे दो तो 50 बार तक डिवाइड कर लेगा, है ना? लेकिन कैंसर सेल लोगे तो कभी नहीं मरेगा, हमेशा के लिए रहेगा. So please learn the 3 type of the cell culture: primary, diploid, and continuous, and understand the differences between them. है ना? Understand. You have to learn the examples. And a primary key, not very important. If you can't learn, it's okay. And a monkey kidney cell, human amniotic cell. Here we are taking the cell of that. We take a monkey, kill the monkey, take the kidney out, and we are taking a monolayer of the kidney cells. Here we are taking human amniotic cell. We take a chick embryo, kill it, and take the cells. And like that. But in diploid, we have two options: WI38 and HL8. WI13 and HL8. I don't know what we are taking, but we are adding growth factors here. And in continuous, there are many cells. Question come on the examples like HeLa, Hep2, McConkey, Viro. So we take cancer cell. Like in HeLa, we take the larynx, monkey larynx carcinoma. Monkey का larynx का carcinoma cell है ना. Hep2 में we take human के cervix का cancer cell. So they all are cancer, but different organ, different animal. But the point is that they all are cancer cell. Say yes, people. Say yes. So what are the three ways to cultivate a virus? I want to cultivate a virus. How should I do so? You will see now the rule is that viruses divide only in living cell. So you have to provide living cell. So I will say, okay, let's provide a living cell. What are the three ways? You will see, ma'am, you can take an animal like mice, है ना? Like mice and inoculate inside that. Or else you can take an egg. There are four membranes in the egg. Chorioallantoic membrane, amniotic cavity, है ना? And yolk sac. You can inject your specimen in any of them. They have the living cell and virus will multiply. Or else you can use the cell lines, cell culture. है ना? There are three type of cell culture: primary, diploid, and continuous. Okay, G, got it, everyone? Say yes, respond. Okay, read the question, tell me the answer. Hep2 is an example of which cell line? Is the primary cell culture, diploid cell culture, continuous cell line, or explant? Yes, what is the correct answer? People, what is the correct answer? Respond. Please respond. Yes, so where I taught you Hep2, continuous. So please learn the examples of continuous. HeLa, Hep2, McCoy, Vero. In सब पे PYQ है. So please learn it by heart. Okay, so we are done. Let me check. No. Inclusion bodies are there. So before going to the inclusion bodies, let me tell you something. Only I can change my life. No one else can do it for me. I hope you all can understand that. I can teach you, I can give you the notes in the simplest version, I can build your concepts, I can tell you how to study, but ultimately, you have to study, you have to win the war. Hannah, you have to do your war. Hannah, I cannot go and give exam in your place. You have to give your own exam and you have to win. Not only this exam, this is not the end, this is the beginning actually. Once you get the license of FMG ना तब तो पिटारा खुल रहा है अभी है ना. तुम्हें लग रहा है पिटारा बंद हो रहा है लेकिन एक नई दुनिया में जाओगे. A new world which you can't imagine. You are entering in that world. You got my point? You are just entering in such a beautiful world where people worship you like God. Literally, see the word God, G-O-D, God. Listen, people will worship you like God. Believe me, you are going to become doctors. I mean, you are doctors, but once you get the license, you start practicing. Whether you do your MD, MS, or PLAB, or whatever exam you are clearing, ultimately you are going to become doctors, great physicians. The first thing you should decide whether you want to be a physician or whether you want to become a surgeon. Hannah, the person cannot be good at both. One person. Either you are good at stetho or you are good at scalpel. You cannot be good at both. So identify yourself, what you want to become, what you want to become, what you are born for, what you are born for. Let me tell you what you are born for. Let me tell you what is the purpose of your life. You are born to save lives, to save lives. This is very big line I have written for you. You are born to save lives on this earth. Only apart from you, the God can do so. You are not God, not even I. No one is equivalent to God. So He is the supreme power. Whosoever the supreme power we worship. Okay, whatever. But after that, second to God, people worship the doctors. Literally, I'm saying. Imagine you are coming out of ICU. Someone is very serious, critical in the ICU. You are coming out of that, and the relatives are waiting, and you are saying, "Don't worry, your dear one is safe. Don't worry, I'm there now. I'm your doctor. Just hold the hand and see the happiness in their eyes." Just imagine you are coming out of OT and saying the surgery is successful. Don't worry. And see the happiness in the eyes of the people. Listen, they can do anything for you. They can, literally. There are no words to explain such feelings. These are the feelings that can be felt, that cannot be said. I'm telling you these feelings. I have felt all these things during my residency, during my working hours in the hospital. We have felt these things. You will also feel once you will start practicing. Hana, imagine you are treating someone in the OT or wherever, and the person is dead. Means we are not God, of course, we cannot save everyone, but we try our best. The person is dead. So, and you are giving this information to the relatives. So, how sad, how the moment it is to be for you also and for the relatives also. The person, no, we cannot save. I'm sorry to say. So these are the very, many things. Sometimes you become emotional in your career, and ultimate purpose to increase two things in people's life: the quality, the quantity. You are born to increase the quality and quantity in people's life, even with your skills, with your knowledge. Hannah, whatever I'm teaching you now, whatever you will study, whatever you will learn in your residency, whatever residency you will do, Hannah, after this, you will give some NEET, PG, FMG, INICT, whatever exam you will give, lab, USMLE, ठीक है, जो भी दोगे, ultimately you will hold some degree. So with your skills and with your knowledge, even if you are increasing one day in someone's life, your life is worth. Your knowledge is worth. You are increasing one day now in someone's life, with your skill. If you have not that skill, no knowledge, so that day was not there. The person was dead one day before. Even if you are increasing one day, your life is worth. Your life is worth, believe me. And you can increase the quality in the years. Ultimately, we have to reduce the suffering. That is what we are for here, the doctors. Got it? Okay. Chalo. Anyways, let's move ahead. Hannah, chalo. Chalo, chalo, chalo. Okay. So let's move ahead. Hannah, I want to motivate more every time. That's why I put many quotes in between. Whenever my PPT runs, you may have seen many quotes, but I don't stop at the quotes. I just move on most of the time because time is always limited. So if I stop somewhere, it will take 2 to 3 minutes, but the syllabus is such a vast thing now. We have to complete that also. Okay, we will make a balance. Don't worry. So let's move ahead. Anna. So ultimately, for that privileged thing, you will have doctor in front of your name, whatever is your name, doctor in front of your name. It's not only a prefix. It's not only a prefix. Anna, it's a supernatural word. People are going to worship you like a superheroes. And believe me, it's not simple prefix. Got my point? So having this word before your name is a great privilege. And for that privilege, you have to do the hard work. गॉड बनना थोड़ी आसान है. गॉड बनाएंगे तुम्हें, आसान थोड़ी है, मेहनत तो करनी पड़ेगी. ठीक है, चलो, let's move now. So let's come on the next topic, inclusion bodies. Let's come on the inclusion body. ठीक है, I guess everyone have heard the word inclusion body. सुना तो सबने होगा, सबने सुना होगा. What is inclusion body? But समझ में कितनों को आता है? What is inclusion body? How many of you know the meaning of the word? Can you see this diagram? I've already explained you the replication of the virus. Can you see this point? The virus is entering. Just a second. This point, the virus is entering. Can you see the virus? Say yes. Can you see this point? The virus is exiting out. Can you see the virus? Yes. What about during this period? You will remember virus is invisible. You told us this is eclipse phase. This is eclipse phase and virus is invisible. I agree. I agree. Now, what if I see this cell under microscope during this time? If I see this cell during light microscope during this time, the virus is not there inside the cell. Actually, the capsid is separated, the nucleic acid is separated, all the things are getting replicated. So I can't see the virus. I can't see the virus, mind my words, but I can see some dot-like structures. Some dot-like structures inside the cell. These dots are known as inclusion body. These dots are. So whenever you get inclusion body inside any host cell, it means that cell is infected with virus. And virus is multiplying inside that cell. Say yes, you got the meaning of that. Say yes, समझ में आया? You got the meaning. What is inclusion body? Inclusion bodies are present inside the host cell. This is a host cell inside which we can see, this is the nucleus of the host cell, inside which we can see the dot-like structures. These dot-like structures are inclusion body. It means that this cell is infected with virus and virus is multiplying inside the cell. It indicates that inclusion body. Hannah, now there are three types of inclusion body. Let me draw. So let me draw the three host cell. This is a host cell, this is a host cell, this is a host cell. Nucleus, nucleus, nucleus. The inclusion body can be in cytoplasm. It's known as intracytoplasmic. The nucleus inclusion body can be in the nucleus. It's intranuclear. And the inclusion body can be both. Hannah, so there are three types of inclusion bodies. What are the three types? Say intracytoplasmic, intranuclear, and both. These are the three types of inclusion bodies we have. Intracytoplasmic inclusion bodies are 5 types. You have to learn the 5 examples. 5 examples here. Intranuclear, again, we have 5 examples, 3 plus 2. I will tell you 5. And here we have 2 examples. So 5, 5, 2. I want you to learn 12 examples at the tip of your tongue. At the tip of your tongue. Okay? Please learn. Yes, viruses don't have the chart. You have to learn from the theory itself. Viruses chart, you can make yourself. Okay? Chalo. Listen, you have to learn. And believe every year one question comes from infusion body. Every year, almost everywhere. It is 5-star, nahi 10-star important topic. Make 10-star. Very important topic. You have to learn the examples. So there are 3 types: intracytoplasmic, intranuclear, and both. So 5 examples here, 5 here, 2 here. So learn the 12 examples. I know it's rectification, but you have to learn. There is no escape. Okay. So what are the 5 types of the intracytoplasmic? Intracytoplasmic के 5 types क्या हैं? What are the 5 types of the intracytoplasmic infusion body? Listen. Have I taught you the DNA viruses? I mean the classification. There are 6 families: double H, A, triple P. Who is the grandfather? The grandfather is the pox. The grandfather is the pox. Pox is a family inside which many members are there. है ना? So these are the 4 members of the pox family. ये 4 तो pox हैं. Out of the 5, 4 are pox. 4 of them are pox. है ना? 4 of them. So vaccinia, variola, fowl pox और molluscum contagiosum. These are the 4 members. You can check. जहाँ पे classification लिखा है ना, you can go to the classification and check the members of the pox. These are the members of the pox family. So you can learn in the cytoplasmic, out of 5, 4 are pox only. 4 are the pox family, है ना? And 1 is rabies, which is the RNA virus. So please learn the name of the viruses first. First learn the name of the virus. 1 is rabies. You have to learn rabies, the dog virus, है ना? Rabies, the RNA virus, and 4 pox virus: vaccinia, variola, fowl pox, and molluscum contagiosum. Please learn the full name. Now you have to learn the name of their infusion body. It is based on the scientist who discovered it. For example, in rabies, the bodies are known as Nigri body because it is discovered by Nigri. In vaccinia, it is known as Guinneri body. In variola, it is known as Paschen body. In fowl pox, it is known as Bollinger body. In molluscum contagiosum, it is known as Henderson-Peterson body. So you have to learn that. So can we revise it together? What are the 5 viruses? Intracytoplasmic. So first tell me the name of the virus. You will say, ma'am, the name of the virus is rabies, and 4 of them are pox viruses. What are the pox viruses? 4 of them. What are 4 pox viruses? Variola, vaccinia, fowl pox, and molluscum contagiosum. Now tell me the name of the infusion bodies, the scientist name. Here Nigri body, here Paschen body, here Guinneri body, here Bollinger body, and here Henderson-Peterson body. Okay, please learn to karna padega. There is no escape. So that. Now coming on intranuclear. The intranuclear are of 2 types: Cowdry A, Cowdry B. You may be thinking, ma'am, what is the 2 type? Ye kya hai? Cowdry A, Cowdry B. What does it mean? What does it mean, Cowdry A, Cowdry B? Okay, let me draw. So let me draw host cell. Nucleus. So here also inclusion body is inside the nucleus. Here also inside the nucleus. But here it's granular. See the granularity. And here it's compact. It's circumscribed. Compact and circumscribed. So the granular one and the circumscribed one. So granular one is known as Cowdry A and circumscribed is known as Cowdry B. Mind my words. Both are intranuclear. Both of them are intranuclear. So please learn that. Cowdry A, Cowdry B. So 3 are Cowdry A and 2 are Cowdry B, total 5. Cowdry A, Cowdry B. So Cowdry A, the mnemonic is heavy, HVY, heavy. And Cowdry B में the mnemonic is PA, heavy PA, HVY, heavy, PA, PA. Herpes simplex virus, varicella zoster virus, yellow fever. H for herpes simplex, V for varicella zoster, the chickenpox virus, and Y for yellow fever. And PA is poliovirus, adenovirus. Got it. Herpes simplex, it is known as Lipschul's body. And yellow fever, it is known as Torrey's body. There is no peculiar name here. जहाँ पे है वहाँ याद करो, जहाँ नहीं है वहाँ नहीं है. So please learn the 5 intranuclear. Learn heavy PA, and both. Finally coming on both. Only 2 examples: measles and cytomegalo. One is measles, one is cytomegalo. In measles, it is known as Waardenfink-Kedle. Waardenfink-Kedle body. And in cytomegalo, it's owl eye appearance. Ullu ki aankh. Owl eye. Can you see? It's looking like owl eye. See the inclusion body present inside the nucleus also and in the cytoplasm also. It's looking like owl eye. Owl face. Say yes. Got it? Got it. Can you help me, someone, in revising inclusion body? You can understand what is inclusion body? समझ में आया क्या होता क्या inclusion body? Huh? So tell me 5 examples here, 5 here, 2 here. Everyone with me. Everyone will say. Okay. So 5 here. 1, 2, 3, 4, 5. Tell me the viruses first. पहले वायरस का नाम बताओ, बाद में इंक्लूजन बॉडी बताना. पहले वायरस का नाम बताओ. क्या है 5 वायरस के नाम? How I learn, it is like 1 is rabies and 4 are pox virus. तो pox virus कौन कौन से हैं? Variola, Vaccinia, Foulpox and Molluscum contagiosum. ठीक है? Now tell me the name of their inclusion body. In rabies, it's known as Nigri body. In variola, it's known as Paschen body. In vaccinia, it is known as Guinneri body. In foulpox, it's known as Bollinger body. And molluscum contagiosum, Henderson-Peterson body. Please learn. Please learn. There is no escape. Learn to karna hi padega. Don't learn, I will slap you. Learn it. Because if you are not learning now, basically you are losing one MCQ out of the 300 in your exam. This is short question. आना ही आना है इसमें से. इतने से पेज में से one question is short. So why to leave that? है ना? Leaving it is foolness. Don't leave that. Okay. Intranuclear is of 2 types, Cowdry A and Cowdry B. They are of 2 types. I told you what is Cowdry A, Cowdry B. Cowdry A is granular and Cowdry B is circumscribed. Both are intranuclear, but one is granular, one is circumscribed. So here I told you mnemonic. 3 are here and 2 here. So I told you the mnemonic heavy PA, HVYPA. है ना? Full form. The H is herpes. है ना? This is varicella zoster and Y is yellow fever. Yellow fever. P is polio and A is adeno. So here names are not everywhere. Here we have few names. In the herpes, it's known as Lipschul's body. And in yellow fever, it is known as Story's body. No other names are there. Coming on both. In the both, we have only 2 examples. Measles and cytomegalovirus. Measles and cytomegalovirus. In measles, it's known as Waardenfink-Kedle body. And in CMV, it's known as owl eye appearance. Please don't dare to forget. Please learn it completely. Okay. So read the question and tell me the answer. Everyone with me? Can you please read the question and tell me the answer? Yes, G. What is the correct answer? Both intranuclear and cytoplasmic are present in only in 2 virus. What is the correct answer? Yes, of course, the correct answer is measles. So measles and CMV. These are the 2 viruses in which both are present. CMV is not there in the option, so we will go with measles. Okay. So correct answer here is measles. Coming on the next, Lipschul's bodies are seen in. You tell me, Lipschul's body kahan padhaya maine? Is it Hodgkin's disease, viral hepatitis, herpes or influenza? I told you heavy pa mein jo H hai, herpes. Herpes mein it is known as Lipschul's. Herpes mein it is known as Lipschul's. So correct answer here is herpes. You all are right. Yes. The next question is Bollinger bodies are seen in. Bollinger bodies. What is the correct answer? Bollinger bodies are seen in. Yes. Bollinger bodies are seen in foulpox. Very correct. Bollinger are seen in foulpox. You all are right. Foulpox, no? Yes, Bollinger are seen in foulpox. I'm changing the question. Instead of Bollinger, I'm giving you Paschen. Paschen bodies. What is your answer now? Paschen body. What is your answer now? Paschen कहाँ होती है? You will say, ma'am, Paschen body are seen in variola. Variola ही smallpox है ना? I will teach you. Variola causes smallpox. In that case, answer will become C. Not A, not chickenpox. It will be smallpox. Variola smallpox. I will teach you. Okay, in that case, answer will become C. The next question, Cowdry type A is seen in which virus? What is the mnemonic? Cowdry A का क्या mnemonic है? What is the mnemonic of Cowdry A? What is the mnemonic? Heavy. Cowdry A is heavy. Cowdry B is PA. है ना? So heavy. Heavy का H. What is H? HBV or herpes? So answer is herpes. Answer is herpes, है ना? And if I change the word A to B, Cowdry B, from the same four options, what is the name? What is the answer? What is the answer in that case? From the same four, Cowdry B के लिए, what is the answer? In that case, pa का A is adeno. ठीक है? Yes, the answer will become C in that case. So you get many questions on that. We are done with general virology. So I told you entire general virology, and now we will start specific viruses. So you know how many DNA viruses are there. I'm not revising it completely. You know all the RNA viruses, all the DNA viruses. है ना? You know here we have 6 families, here we have 15 families. अब तो रट गया होगा. You know all DNA viruses are double-stranded except one. All RNA viruses are single-stranded except one. And you should know these exceptions. You know who's largest, smallest in these categories. Who is enveloped, who is naked, who is enveloped, who is naked. Learn the naked in both categories. Here naked is PAV, here naked is PCR. Remaining will fall in enveloped category. You may be knowing the symmetry of them. You may be knowing who is segmented. You may be knowing the ways to cultivate and inclusion body. Now let's start viruses one by one. Can I start the marathon, the viruses? So the plan is that I will teach you 6 DNA viruses, all 6, one by one. All 6, one by one. And then I will teach you among the RNA, the 15, I will teach you 6 or 7 here. I know the important 6, 7. We will not study all, but the important we will cover. So can I start the DNA? I'm starting with DNA. Let's start with the DNA viruses, the 6 viruses. Anna, 6 viruses. The first virus I'm starting with pox. The first I'm starting pox, one of the DNA virus. I will not start pox. First you tell me what do you know about pox. पहले तुम बताओ तुम्हें क्या-क्या पता है. Write down. क्या-क्या पता है pox के बारे में. बहुत कुछ you know. Whatever you know, kindly let me know. Then I will start it. What do you know about the pox? You will see, ma'am, it is largest. Very good, Elizabeth. Elizabeth, it is largest what? See, ma'am, largest DNA and largest overall. दोनों चीज बोलो. Largest DNA भी है और largest overall भी है. ठीक है. और क्या पता है pox के बारे में? इसकी symmetry बताओ चलो. What is the symmetry of pox? हाँ? Is it single stranded or double stranded? वो बताओ. DNA तो है, but single or double? हाँ? Symmetry is what? All the DNA viruses are icosahedral except pox. The pox is complex. याद आया क्या? Pox is complex. ठीक है. Single stranded है कि double stranded है? सारे DNA viruses are double stranded except parvo. ये parvo तो है नहीं. So it is double stranded. It is double stranded. है ना? इसका shape बताओ. What is the shape? I told you the shape of the pox. The shape of the pox, I told you brick shape. It is brick shape. और बताओ ये enveloped है कि naked है? You tell me. Enveloped है कि naked है? DNA viruses में ये grandfather है. Grandfather को respect दोगे नहीं दोगे? Would you give respect or do not give respect? It is enveloped. So ये सारी चीजें you already know, है ना? From your mnemonics and all. ठीक है ना? See, the same thing is written. The first page, nothing extra. You know everything. Say yes or no. So the shape of the pox virus is brick. You know that? Yes, you already know. It is enveloped. It is the grandfather. We will give the respect. We know. Symmetry is complex. It is an exception. Rest all DNA viruses are icosahedral. This one is complex. This is the largest DNA and largest overall. Largest DNA as well as largest overall. ये सारी चीजें पता है. No need to learn. You already know that. Why to learn? You already know that. है ना? The point which is additional here, you can see the capsid is dumbbell shape. The capsid is dumbbell shape. Dumbbell shape का core है ना? Dumbbell shape. And around the dumbbell shape, 2 lateral bodies are there. है ना? So typical diagram, brick shape you can see. So dumbbell याद करो. In this family, there are many members. There are many members, but I will not teach you all. I will teach you 3, which cause disease in human. The remaining cause disease in animals. तो वो veterinary doctor पढ़ेंगे. हम नहीं पढ़ेंगे. है ना? हम कौन से 3 पढ़ेंगे? The 3 poxviridae member के 3 member. One is variola, one is vaccinia, one is molluscum contagiosum. One is molluscum contagiosum. That's it. I will teach you only 3. Variola, vaccinia, and molluscum contagiosum. है ना? So I will teach you only 3 viruses. Variola, vaccinia, and molluscum contagiosum out of all these. But you should know it is having many viruses. समझ में आ रहा है क्या? है ना? So I will teach you only 3. Variola, vaccinia, and molluscum contagiosum. है ना? One by one we will see, and this chapter will be over. I've started the first family, poxviridae. है ना? Before classification, I told you the diagram. So this is a diagram for all of them. whether it is variola, vaccinia, mollusca, disease, the diagram is same. You know the morphology, and now you know the classification. Now 3 we will see one by one in detail. Let me start with the first one. The first one is the variola. Variola is a causative agent of smallpox. Have you heard about the disease smallpox? This is smallpox. सुना है smallpox के बारे में? This is smallpox. It is the only disease which is eradicated from the world. Till date. एक ही disease को हमने eradicate किया है. Eradicate from the world. Till date. What is the date? पढ़िए PSM में. It's 8th May 1980. It's 8th May 1980. It was eradicated from the world. After that, none of the case till date. आज तक एक भी case नहीं मिला. Got my point? So that is the only disease which is eradicated, smallpox. So let me draw the pathogenesis. Let me draw a human being. You tell me how does this virus enters human body? I'm asking about the pox virus ka variola. Variola. How does it enters in human body? Variola. How does it enters in human body? It enters via nasal route. I mean droplet. It enters, it enters via droplet. After entering from the droplet route, it goes to the lungs. After going from lungs, it will go to the blood. After going from blood, it go to only one tissue, the dermal tissue. This is dermal tissue. This is skin, dermal tissue. So it will go only to the dermal tissue and produce lesions there. And the disease is known as smallpox. You can see. This is smallpox. This is smallpox. So the virus is inhaled, lung the root is inhalation. After that it goes to the lung. From there it goes to the blood. From blood it goes to the dermal tissue and produce pox. The lesions are known as pox. P-O-C-K-S. These all chote lesions are known as pox. All these lesions are known as pox. Do you have any confusion? All these lesions are known as pox, variola virus. है ना? The name of the disease is smallpox. So smallpox में क्या होता है? You can see. Please everyone on the screen. The rash starts from the mouth. The rash starts from the mouth and spread to the periphery. Like this. It spread to the periphery. It is going away from the center. That's why known as centrifugal. Away from the center. The rash starts from the mouth and yes, including palm and sole, it spread to the periphery. है ना? So it spread to the periphery. The rash is known as centrifugal. The rash is known as centrifugal. Please learn that. है ना? Now, if you rupture one of the vesicle, take one of the vesicle and rupture it. If you rupture it from inside, you will see it's unicompartment. I'm sorry, it's multi-compartment. Unicompartment वो वाला होगा. This one is multi-compartment. This one is multi-compartment. It's very deep. It's very deep. है ना? It's very deep. So please learn these findings. Only one organ is spared, axilla. There is no rash in axilla. It is there on palm, sole, but axilla is spared. Axilla spared. And now, yes. So that is centrifugal. Of course, it starts from mouth and spread everywhere. Not only palm, soles. It spread everywhere on the body. You can see in the diagram also. No, this is centrifugal. Okay, got it. Now there is one more disease, that is chickenpox. Have you heard about chickenpox? Chickenpox हुआ है किसी को बचपन में? It's very common. मुझे भी हुआ है. So chickenpox is very common. It is not eradicated. It's very common. So what is chickenpox? I'm telling you two things. One is chickenpox. One is smallpox. I know both of them is caused by DNA virus. This is also a DNA virus. This is also a DNA virus. I know here the name of the DNA virus family is poxviridae. Here the name of the DNA virus family is herpes. ठीक है? Here poxviridae में कौन सा member है? Variola, which causes smallpox, which I'm teaching you right now. यहाँ पे chickenpox कौन कराता है? Varicella zoster, that causes chickenpox. So why we are comparing the two? दोनों का रैश सेम है. दोनों डीएनए वायरस से होते हैं, लेकिन अलग अलग वायरस हैं. फैमिली अलग है, वायरस अलग है, लेकिन रैश इज सिमिलर. दिखने में एक जैसा है. The rash is similar. So let me compare the rash of the chickenpox with smallpox. You get many questions from this. Please see, everyone on the screen. This is smallpox. This is chickenpox. Everyone on the screen. Smallpox rash starts from the center. It starts from the center and spread to the periphery like this. It spread everywhere to the periphery like this. It is centrifugal. On the contrary, chickenpox rash start from the periphery and come towards the center. Start from the palms and soles and come towards the center. This is towards the center, so centripetal. How you will learn? You will forget in the exam कि कौन सा centrifugal है, कौन सा centripetal है. कैसे याद करोगे? So I learned CP for CP, centripetal for chickenpox, CP for CP. है ना. Chicken pox, centripetal, you will not forget now. CP for CP, you get many questions on that, है ना. So yes, okay, so this is the rash. So one is centripetal, one is centrifugal, you have to learn that. उनमें से एक centripetal है, one is centrifugal. Now learn one organ which is paired in both of them. In smallpox, I already told you the axilla is paired. In chickenpox, the palms and soles are paired. Learn the organ of spearment. One organ is paired. Come on the rash. Come on the rash. Can you see chickenpox? Can you see smallpox? In chickenpox, can you see some of them is papule, pustule, macule. Some of them is macule, papule, pustule. So they all are together. है ना? And here you can see they all are vesicle. They all are vesicle. है ना? Either all of them will be macule, all of them will be pustule, all of them will be vesicle. So my point is that in chickenpox, you get various stages at the same time. That's why rash is pleomorphic. Rash is pleomorphic, but in smallpox, one stage at a time. The rash is monomorphic. Monomorphic and synchronous. The rash is synchronous. Monomorphic and synchronous. Say yes. So please understand. This is pleomorphic. This is monomorphic. Hana? So that is a rash. You can see this is a rash. Got it? One more thing you have to understand it here. In chickenpox, around each rash, you can see the redness. Around the rash, appreciate the redness. So here inflammation present. Here inflammation is absent. Hana, here I can see only rash. Around the rash, I can see nothing. Here inflammation present, here inflammation absent. Got it? Please learn inflammation present, inflammation absent. Okay, got it? That is the thing. And last point, if you rupture the rash, if you rupture one of the vesicle, both places, rupture, यहाँ भी rupture करो, यहाँ भी. If you rupture here, it is unique compartment. If you rupture here, this is multi-compartment. This one is superficial, this one is deep. How many of you got it? You have to learn the differences. You have to learn the differences. Can we write the differences? You will tell me. Chicken pox, smallpox. ठीक है? First tell me which is centripetal, which is centrifugal. Learn CP for CP. Centripetal, centrifugal. Centripetal, chicken pox. This one is centrifugal. ठीक है? Please learn that. After that, second thing you will learn. क्या बताई थी second thing? Centripetal, centrifugal के बाद both of them rash start करो. Here rash is pleomorphic. Here rash is monomorphic. है ना? Here rash is superficial. Here rash is deep. Here rash is unicompartment. Here rash is multi-compartment. Here inflammation present. Here inflammation absent. Any other point? So please learn the differences. Please learn the differences between the rash of chickenpox and smallpox. I know. So same thing is written. One organ is paired. You have to learn. So here in chickenpox, the palms and soles are paired, and in smallpox, the axilla is paired. Please learn the organ of spearment of both of them. I know. So that is the rash. So we are done with variola. I have to teach you 3 viruses now. Variola, vaccinia, and molluscum contagiosum. So first chapter will be over. I have to teach you 12 chapters. मैं पहला पढ़ा रही हूँ अभी तक, है ना? 11 more to go. So variola is done. Now coming on vaccinia. Coming on vaccinia. नाम में ही है. This is not a natural virus. This is not a natural virus. This is an artificial virus. It is used to make the vaccine. Vaccine. That's why vaccinia. I don't know when you all are born. All the people who are born after 1980, they are not vaccinated for smallpox because government has discontinued the vaccine. क्योंकि 1980 में the world is declared free of smallpox. तो वर्ल्ड में जब वायरस ही नहीं है तो उसका वैक्सीन लगा के क्या करोगे? So we have stopped the vaccines. लेकिन ask your parents, ask your grandparents, है ना? They may be knowing this disease and they all are vaccinated because they are born. By the way, I am also born after 1980. You may be thinking कि मैम तो 1980 की पहले की होगी. नहीं, है ना? I am also 1980 का after ही. ठीक है? So जितने भी लोग 1980 के बाद के birth है ना? So I am also not vaccinated for smallpox. So all the person who are before 1980 born, so they all are vaccinated for the smallpox. So ask your parents, and my parents are vaccinated. Ask your grandparents, and they will show you the mark. Sometimes they remember. Or na, they call it in Hindi chota chechak. The disease is known as chota chechak, smallpox. They call the pox as chechak, the disease. It was a deadly disease at that era. Hana, they get afraid for that, and they are vaccinated for that. Hana, but after 1980, the vaccine is discontinued. Okay, got my point. So after 1980. So at that point, before 1980, जो वैक्सीन बनता था वो कैसे बनता था? Before 1980, जो वैक्सीन बनता था वो कैसे बनता था? वो वायरस से ही बनता है ना? वैक्सीन बनाते कैसे हैं? How do you make वैक्सीन? वैक्सीन कैसे बनाते हैं? वी टेक द वायरस, है ना, एंड वी एटीन्यूएट इट. एटीन्यूएट, यू नो व्हाट इज एटीन्यूएशन? एटीन्यूएशन मतलब वीकनिंग. उसको वीक करके अंदर डाल देंगे. वी विल इंजेक्ट इट. So virus will not cause the disease because it is attenuated, but it is antigenic. But the virus is deadly, na? Imagine some person is making the vaccine और उसी को exposure से smallpox हो गया तो. So what we have done, we have taken the variola virus, है ना, and we have changed one antigen in that. Changed only one antigen. So the virus which is formed is vaccine. Vaccinia. So vaccinia is only variola. It's an artificial virus. It's an artificial virus. It's not natural. It's artificial virus. It is formed by changing one antigen in the variola. We changed one antigen in the variola and it resulted in forming vaccinia. So vaccinia is safe. Vaccinia do not cause smallpox. This cause smallpox. This do not cause smallpox. One more difference between them. Can anyone tell me? One more difference between them. Huh? Variola की bodies, inclusion body, Paschen body और यहाँ पे Guinneri body. Tell me the differences between the two. So I have taught you the two viruses till now. This is variola. This is vaccinia. Say, ma'am, this is natural virus. Say, ma'am, this is artificial virus. Say, this is pathogenic. It causes the disease. And this is non-pathogenic. It do not cause the disease. है ना? It causes a disease known as smallpox. But this one is used for vaccination. है ना? The inclusion bodies are different. Here we have Paschen body. And here we have Guinneri body. Make some mnemonic कि वेरियोला में पास्चन है और वैक्सीनिया में गुइनेरी है. So you will not confuse. वैक्सीनिया में गुइनेरी bodies है. So you have to learn that. याद हुआ क्या? समझ में आया क्या? Anyone have any confusion? The last one is molluscum contagiosum. I will teach you 12 viruses today. Now there are many viruses, 6 DNA, 15 RNA, many viruses. है ना? Till date only one virus cannot be cultivated. We have 3 ways to cultivate the virus. Either mice, that is animal inoculation, या फिर egg, है ना, या फिर सेल कल्चर, सेल लाइंस, है ना, द प्राइमरी डिप्लॉयड कंटिन्यूअस, है ना. मॉलस्कम कंटेजियसम कैन नॉट बी कल्टिवेटेड इन एनी ऑफ देम टिल डेट. हम उसे कल्टिवेट ही नहीं कर पाए. नाइदर इन द एनिमल नॉर इन द एग नॉर इन द टिश्यू कल्चर. नॉन कल्टिवेबल वायरस. नॉन कल्टिवेबल वायरस. प्लीज हाईलाइट एंड लर्न दैट. प्लीज हाईलाइट. It causes a disease. The name of the disease is molluscum contagiosum. Here also the person have rash. But there is a peculiarity in the rash. हर rash के center में hole है, depression है. Every rash, can you see all the rash? This is a rash, this is a rash. All the rash have a depression at the center. Can you appreciate the depression at the center? This is known as umbilicated rash. What is known as umbilicated rash? I know. And inclusion body is different in all of them. So we are done. I'm teaching you virology. I'm teaching you DNA viruses. I have to teach you 6 DNA viruses. I'm teaching you first one only. Pox. In the pox, I told you the morphology. है ना? It's a brick shape. Brick shape का होता है. This one is the largest virus. Largest DNA virus. Largest overall virus. है ना? And here we have a dumbbell capsid and 2 lateral bodies. ये तो diagram हुआ. इसके अलावा I taught you 3. 1, 2, 3. 3 type of viruses: variola, vaccinia, and molluscum contagiosum. है ना? Here I taught you this causes a disease smallpox, which you have to differentiate from chickenpox. ठीक है? Here it causes a disease smallpox that you have to differentiate from chickenpox. We have seen the differences. Vaccinia is an artificial virus. It's not natural. It is used for vaccine. It is non-pathogenic. And coming on the molluscum contagiosum, here the rash is umbilicated. Number 1. And number 2, this is the virus which is not cultivated till date. Give me a thumbs up. I'm done. I'm done. देखो मैंने पूरा चैप्टर एक पेज पे समराइज कर दिया. Poxvirity मैंने कंप्लीट किया. I taught you 3 things there. 3 viruses: variola, vaccinia, molluscum contagiosum. Respond, please respond. Can we go ahead? Read the question. Tell me the answer. Quick. Which virus cannot be cultivated till date? आज तक सिर्फ एकमात्र वायरस which we cannot cultivate in none of the media. No. Neither in animal, nor in tissue culture, nor in egg. Yes, the correct answer is molluscum contagiosum. Please learn that. Guinneri bodies are seen in. कहाँ पे होती है Guinneri? Guinneri bodies. Yes. What is the correct answer? Yes, of course, the correct answer. Guinneri bodies are seen in vaccinia. है ना? I'm changing the question. I'm changing the question to Bollinger bodies. What do you say now? Do you like to change your answer or still same? Would you like to change your answer or still same? I'm asking. Bollinger body, they are seen in foul pox. You all are right, they are seen in foul pox. And if I change the question again, Henderson-Peterson body, Henderson-Peterson, everyone knows the answer. It's molluscum contagiosum. So we keep on changing the question, the answer will change. Okay, the next question in front of you. Don't jump on the answer. Read the question and understand it. There is a child, take the clue. Hana, there is a child presented with umbilicated lesion. The word to be picked here is umbilicated lesion. The image is also provided to you. So can you see? This is the lesion, and you can see the central depression. So of course the answer here is a molluscum contagiosum, but it is not given in the option. क्या मार्क करोगे? molluscum contagiosum नहीं दिया, लेकिन उसकी family तो दी है. The family is pox. The family is pox, so answer is pox. How many of you got it? So can we proceed? आया समझ में? So we are done with one of the DNA virus. 6 में से एक हो गया. Can I move to the next one? The next one is the second virus, parvovirus. First you tell me. अभी हमने grandfather दे दिया, देख लिया, आप baby को देख लेते हैं. So let's start parvo. You tell me क्या क्या पता है parvo के बारे में. Let me ask, चलो क्या क्या पता है? इसका size बताओ. इसका साइज बताओ, स्मॉलेस्ट है कि लार्जेस्ट है ना, उस पर कमेंट करो. इसकी सिमेट्री बताओ, है ना, ये सिंगल स्ट्रैंडेड है कि डबल स्ट्रैंडेड है वो बताओ. ठीक है, ये इन्वॉल्व्ड है कि नेकेड है वो बताओ. ठीक है, ये 4 चीजें बताओ. यू टेल मी. हाँ, यू टेल मी व्हाट इज द साइज. यस, वेरी गुड. स्मॉलेस्ट है, लेकिन स्मॉलेस्ट डीएनए भी है और स्मॉलेस्ट ओवरऑल भी है. राइट टू थिंग्स सेपरेटली, बोथ थिंग्स. स्मॉलेस्ट डीएनए भी है, स्मॉलेस्ट ओवरऑल भी है. बोथ द स्मॉलेस्ट विल कम हियर. सिमेट्री कौन सी है? सिमेट्री कौन सी है? दे ऑल हैव आइकोसाहेड्रल सिमेट्री एक्सेप्ट पॉक्स, जिसकी कॉम्प्लेक्स होती है. सिंगल है कि डबल है? यस, डबल. नो संदेश. It's single stranded now. Everyone is saying double or single. Let me check. It's single stranded virus. I told you all the DNA viruses are double stranded except parvo, which is single stranded. Is it enveloped or naked? It's a baby. Baby can be naked. Baby can be naked. Pap. Pap का one of the P है. Okay, this one is naked. समझ में आई क्या? ये सब लर्न करने की जरूरत नहीं है. This you already know. Parvovirus. Do you know its smallest DNA? Yes. Do you know its smallest overall? Yes. Do you know it is the only virus which is single-stranded DNA? Only. Yes. Do you know this is naked? Because it is baby. Babies are naked. No clothes. So you know everything on this page. No need to learn anything. You already know that. Okay. Let me come on the diseases. It caused 3 diseases. है ना? The disease in the children, the disease in the adults, the disease in the fetus. ठीक है, सबसे पहले चिल्ड्रन की बात करो. कैन यू सी दिस चाइल्ड? यस, ऐसा लग रहा है कि मम्मी ने चांटा मारा. हाँ, मम्मी ने चांटा मारा. इट इज लुकिंग, कैन यू सी द चीक? द चीक इज रेड. इट इज लुकिंग समवन हैव स्लैप्ड द बॉय. सो इट इज नोन एज स्लैप्ड चीक फीवर. व्हाट इट इज नोन एज स्लैप्ड चीक फीवर. व्हाई इट इज अकरिंग? नोबडी हैव स्लैप्ड. किसी ने इसको स्लैप नहीं मारा. Actually, the boy is having the infection of the parvo. This child is having infection of parvovirus, and the parvovirus causes cutaneous vasodilatation of the face. Cutaneous vasodilatation of the face. That's why it is looking red. It is not the slap. It is cutaneous vasodilatation. So it is also known as fifth disease, because after entering the viruses, after 5 diseases, 5 days it presents. It is known as third disease. Okay, and since it become red, it is known as erythema infectiosum. So my point is the 3 things are same. Whether you say slap cheek appearance, whether you say 5th disease, whether you say erythema infectiosum, the meaning is same. And these are the 3 presentation in children. Adult में क्या होगा? Adult में only one thing is there, arthralgia, especially on the wrist joint. Adults में arthralgia, especially on the wrist joint. और fetus में क्या होगा? Fetus में severe anemia, that is hydrocephalus. Severe anemia, that is hydrocephalus. Let me tell you, fetus में, you know, we all have bone marrow. This is the bone marrow of the fetus. Fetus का bone marrow है. In the bone marrow, the first cell is hematopoietic stem cell. That give rise RBC, WBC platelet. सबको give rise करता है. So this is RBC precursor, WBC precursor, and platelet precursor. So whenever this fetus have infection of the parvovirus, this is parvovirus. Parvovirus go to the bone marrow. It go to RBC precursor and kill them. It kill only RBC precursor. It don't kill WBC and platelet. precursor. So patient have anemia. I mean, the fetus have severe anemia known as crisis. It is known as aplastic crisis. Aplastic crisis. And hydrocephalus and abortions can occur. So this is parvovirus. We are done. छोटे छोटे से वायरसेस हैं, फटाफट हो जाएंगे, है ना. सो पार्वो के बारे में क्या क्या पता है? You will comment on the size, you will comment on DNA के RNA, I mean genetic material. You will comment on single stranded या double stranded, है ना. You will comment on symmetry. ठीक है, तो ये सारी चीजें बोलते हुए आपको डिजीजेस बतानी है. The 3 diseases: the disease in the fetus, the disease in adult, and the disease in children. हाँ, size में बोलो it is smallest, genetic material में बोलो it's DNA virus. All DNA viruses are double stranded. This one is single stranded. Symmetry is icosahedral. One thing I forgot to write. This one is enveloped or naked. So answer will be naked. This is the baby. The babies are naked. And I'll learn the 3 diseases mainly. So in fetus, it is aplastic crisis. Aplastic crisis. In the fetus, it's aplastic crisis in which all the precursors of the RBCs are dead. In the adults, it's arthralgia. Okay? And in children, it's slapped cheek appearance. How many of you got it? We are done with this virus. Read the question, tell me the answer. Slapped cheek appearance with the fever is seen with which virus? Slapped cheek appearance. Yes, slapped cheek appearance is seen in which virus? Yes, sometimes congenital heart disease, congenital heart failure can also occur. Yes, of course, the correct answer is parvo, and you all are right. Very correct. Anna, in parvovirus infection, what is common in adult? Read the question. I'm asking in adult. Adult, mind my word. I'm asking in adult. Is it bone marrow aplasia, PARCA, erythema infectiosum, or arthropathy? So let's wait. I'm asking you. What's the correct answer here? Yes. So in adults, it will cause arthropathy. The correct answer is arthropathy. Instead of adult, if I say child, what's the answer now? Child में क्या आंसर होगा? बच्चों में क्या कराता है? स्लैप चीक. लेकिन वो तो दिया नहीं है, तो उसका दूसरा नाम क्या है? एरिथमा इन्फेक्शियोसम. Not A. The answer will be C. Why some people are saying A? In that case, answer will be C. And if I do the fetus, fetus में क्या हो जाएगा? Fetus में हो जाएगा pure red cell aplasia. Only the red cells are destroyed, not others. It is known as pure red cell aplasia या aplastic crisis. So this is how I have given you 3 questions here. समझ में आया क्या? We are done with 2 viruses. Coming on the third one. है ना, 12 में से 2 हो गए, है ना. थर्ड पे आ रही हूँ. I'm coming on the third one. So the third one is adenovirus. What is the third one? The third one is the adenovirus. Start adenovirus. Okay, the third one is the adenovirus. Let's start adenovirus. Adenovirus के बारे में क्या क्या पता है? First you tell me. Then I will start. Adeno. Adeno के बारे में same thing. You tell me. First you tell me genetic material. Then you tell me is it enveloped or naked. Then you tell me is it single stranded or double stranded. ठीक है? What else? You tell me the symmetry. You tell me the shape. That's it. Who will tell? Genetic material is DNA. And is it single or double? Like all viruses, it is double only. Exception is it parvo आएगा. Is it enveloped or naked? पाप का A है ये. पाप का A. So this one is naked. This one is naked. Symmetry is icosahedral, like all other DNA virus. Shape क्या है? Space vehicle. Yes. So the shape is space vehicle. Yeah, satellite shape that you have to learn. है ना? So let's start adenovirus. So most of the things you already know. You already know. See the morphology. Can you see here? Space vehicle. Can you see this is capsid? What we are looking is capsid, inside which nucleic acid is there. But it is covered with a capsid. In the capsid, have I taught you capsid? Capsid is a polymer made up of monomer, which is known as capsomer. The monomer is known as capsomer. The capsid is a polymer made up of a monomer. The monomer is capsomer. You understood this? Hana? So total 252 capsomers are there. 252 capsomers are there. You have to learn that. Hana? 252 capsomers में से 240 are the blue color. Can you see? And 12 of them, they are forming knob-like structures like this. They are forming the knob-like structures like this. The 12, है ना? So 12 are pentones and the 240 are hexons. And because of which It is the space vehicle shape. Space vehicle shape का reason ये है, because of which it is space vehicle shape. So can you tell me, adenovirus में कितने capsid होते हैं? How many capsids are there? So there are total 252 capsid. 240 are hexon, and out of them 12 are pentons. The pentons are forming knob-like structure, because of which it is space vehicle shape. So someone can ask you the total capsid, है ना? You have to learn 252. उसमें split they can ask, है ना? 240 plus 12. 240 plus 12. Learn the split. It's 240 plus 12. Got it? That is adenovirus. How many type of adenovirus are there? 52 type. 52 type, है ना? Type 1, 2, 3, 4, likewise there are 52 type. And these all diseases are caused by adenovirus, है ना? Adenovirus cause all these diseases. Got my point? These all diseases are caused by the adenovirus. You can't learn all, learn the important one. You have to learn the corresponding serotype also. So learn pharyngoconjunctival fever is caused by 3714. It's a PYQ, है ना? Learn the 3 type of conjunctivitis that you will study in ophthalm also. These are the 3 type of conjunctivitis. Okay, follicular conjunctivitis caused by 37. इसी में से 14 remove कर दिया. Acute keratoconjunctivitis is caused by 8, 19 and 37. Acute hemorrhagic conjunctivitis is caused by 11 or 21. You have to learn the serotypes, at least of the important type. We are done with adeno. Adeno में एक ही question आता है, space vehicle. So here one more question, pharyngoconjunctival fever is caused by. What's the correct answer? I told you pharyngoconjunctival fever right now, no? You want to see? So it's 3714. So answer is 37. You all are right. The answer is 37. Pharyngoconjunctival fever. Space vehicle viruses. Which virus is space vehicle? Is it reovirus, rhabdovirus, retrovirus, or adenovirus? Yes. So yes, space vehicle virus is obviously adenovirus. 252 capsid, है ना? 242, 240 plus 12. You all are right. Okay. Can we move to the next virus? So we are done with 3 till now. Total 6 DNA में से 3 are done. 3 more to go. So DNA will be packed, and after that we will come on RNA. It's a small unit. Virology छोटा है, not very big. Parasite थोड़ा बड़ा है ना? But from tomorrow morning itself we will start parasite. The last section, parasito and then myco. Okay. Before coming on the next thing, let me tell you something. I have seen most of the students, they keep on competing with their friends, with their colleagues, with their senior, with their junior. They always give the GTs. I know you people also give the GT. And they always have the scores in the GTs, of course. So sometimes the scores are good, sometimes scores are bad, and they used to compare with a friend. कि उसके इतने हैं, मेरे इतने हैं. No, never do so. Never do so. It's a wrong thing. If you want to compete with someone, compete with yourself. What do you mean by that, compete with yourself? Just suppose in the last GT you got 40% marks. So in this GT, your target is not less than 40. It can be 41, but not less than 40. So this is how you compete yourself. You start competing yourself. Your next performance should be better than the last. Hana? So if you follow this, there is a hyperparabolic pathway, and definitely you will get the success. Got my point? Say yes or no. So listen, never compare yourself with others. Everybody have a different pace. Everybody have a different speed. There are some students who start slow but gradually pick up the pace. There are some students who start very fast but eventually they slow down. Life changes, circumstances changes. The people who are ahead of you, they are not always ahead of you. The people who are behind you, they are not always behind you. So every time it's phase. It's not that this time your rank is 3000, it will always be there. So the remaining 3000 ahead of you and the 10,000 behind of you, the scenario will not change. Got my point? So everything changes. Hannah, have you heard about the hare and turtle story? Huh? तो नहीं है क्या खरगोश और this tortoise का story, कछुए का story? सबने सुना है ना? Consistency is the key. I've seen the student, है ना? क्या करते हैं वो? कि today I have motivated you a lot. आज बहुत motivated हो. आज तो इतना पढ़ेंगे after the class you are keeping studying. You studied for 18 hours today. है ना? 17 hours today. Tomorrow also, a day after tomorrow also, you are burnt out. Finally, you are burnt out, and the next 7 days you don't study at all. Do you do so? तुम करते हो क्या? Don't do that. Don't ever burnt out. No. Maintenance, consistency. Every day just study for after the classes. Mean total, including the classes I am saying, 10 to 12 hours, including your classes, plus self-study. Maintenance. It is not 18 hours. So you will study 18, 18, 18, 0, 0, 0, 0, 0, like this. No, this pattern is the worst pattern. It will lead to you failure. Rather, every day, 10 to 12, 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 10, but it is consistent. Consistency is the key. ठीक है? So can we go ahead now? Huh? So can I start the next virus? The next virus I'm starting here is the Papova. So till now we have seen 3 viruses. I've started pox, I've completed parvo, I've completed adeno. I'm starting the 4th virus, Papova. So let's start Papova. Okay? Papova का नाम Papova कैसे पढ़ा? There are 3 viruses. Pa, Po and Va together constitute Papova. Do you know that? Papova में 3 viruses हैं. The Pa is papilloma, Po is polyoma, and Va is vacuolating virus. I will not teach you Va because it causes disease in animal. हमें नहीं पढ़ना है. हमें Pa और Po पढ़ना है. So I will teach you 2 viruses here. Pa is papilloma and Po is polyoma. So just learn the 2 viruses, Pa and Po. Got my point? Papilloma virus and polyoma virus. One is papilloma, one is polyoma. Papilloma is human papilloma virus. So let me start with human papilloma. It's very important. Let's start human papilloma virus. Yesterday, have I taught you there are certain oncogenic organisms? मैंने बोले थे क्या oncogenic organisms? There is only one bacteria which is oncogenic. You will tell me which one. Yesterday I taught you. There are 7 viruses which are oncogenic and 4 parasites which are oncogenic. Huh? 7, 8, 9, 10, 11, 12. 12 organisms are oncogenic. कल बताया था. Respond. So which, Elizabeth, which bacteria? Adeno तो ठीक है. Yes, yes, very good. Who is this? Arnold. Yes, very good. So correct answer is H. pylori. H. pylori is the only bacteria which is oncogenic. But viruses 7 now. The first is coming now. HPV. I will teach you 6 more. आज ही आएंगे, अभी आएंगे. The first is HPV. HPV virus. Let's start with human papilloma virus. HPV virus. It is an oncogenic virus. It causes cancer. How does it cause cancer? It causes 2 cancers. है ना? Number 1, male and female. It causes genital cancer. In males, it causes penis ka cancer. In females, it causes cervix ka cancer. And it causes anal cancer in both of them. So basically, it is known as anogenital cancer. Anogenital. Ano is same. Genital is different. It is known as anogenital cancer. Anna? So HPV causes anogenital cancer. Let me draw a cell. This is a genital cell of male, female, kisi ka bhi. Anna? This is the human ka genital cell. This is the HPV virus entering. So HPV virus, after entering inside the cell, form 2 proteins. It formed 2 proteins, E6 and E7. It formed 2 proteins, E6 and E7. ठीक है? Both of them move to the nucleus. E6, after moving in the nucleus, it is causing the mutation of P53 gene. And E7 causing mutation of retinoblastoma gene. So because P53 and retinoblastoma are mutated, this cell will become immortal. It will convert into cancer cell. It will convert into cancer cell. कितनों को समझ में आई? How many of you got it? You got my concept, what I told you. So HPV goes to the skin cell, mucous cell or genital cell. So it forms 2 proteins, E6 and E7. E6 inhibit P53 and E7 inhibit retinoblastoma that will lead to cancer. Say yes, got it. Please learn that. It causes 2 cancers, benign and malignant. So HPV causes 2 types of cancer, benign cancer and malignant cancer. It causes 2 types of cancer, benign cancer and malignant cancer. It causes both. The benign cancers are known as warts. And the malignant cancers are known as neoplasia. Okay, so HPV causes 2 types of cancer with the help of E6 and E7 protein. ठीक है, E6 protein mutate P53. You have to learn कौन सा क्या mutate कर रहा है. And E7 mutate retinoblastoma. And finally, it leads to cancer. The benign are the warts and the malignant are the neoplasia. Okay, so this is the story written in front of you. ठीक है, the same story is shown to you. You can see E6, you can see E7. ठीक है, so E6 is inhibiting P53, E7 is inhibiting retinoblastoma, leading to cancer cell. ठीक है, what are warts? What are warts? These are the benign cancer, na? 3 types of warts are there. See in the diagram. If the warts are present on hands and finger, they are known as verrucous vulgaris. If they are present on soles of the feet, they are known as verruca plantaris. And if they are present on the face, they are known as verruca plana. So verruca vulgaris, verruca plantaris, verruca plana. Please learn the 3 type of the warts. And now the next is the anogenital warts. ये तो skin के थे, skin के warts, the cutaneous warts. Anogenital, can you see the vulva of a female? Around the vulva, appreciate the warts. Appreciate the warts around the vulva. Around the vulva, the warts are there. It is looking like cauliflower-like growth. Cauliflower-like growth. Hana, it is known as condyloma acuminata. Please, you get a question on the condyloma. Condyloma acuminata means cauliflower-like growth. Please learn. Anogenital warts are known as condyloma acuminata because of the cauliflower-like growth. And in the larynx also warts are there. है ना? ये सारे benign थे. ठीक है? Now I'm coming on malignant. Malignant में cancers. It causes cancers. Listen. What I'm teaching you right now, I'm teaching you HPV. And how many types of HPV is there? There are 100 types of HPV. We divide them into 2 categories. Low risk HPV and high risk HPV. What is low risk HPV? Low risk HPV causes benign cancer warts. And high risk HPV causes malignant cancers, that is neoplasia. So tell which one is low, which one is high. Low में 2 ही number याद करो, 6 और 11, 6 and 11. Learn that. 6 and 11, they causes warts. और malignant में learn 5, 16. Learn the sequence. 16, 18, 31, 33, 45. है ना? 45 and 58, I guess, or 54. I will confirm. So that is high risk. Please learn. Yes, very good. Very good, Suji. It's 6 and 11. So all the warts I shown you ना, all these cutaneous warts, anogenital, देखो यहाँ भी है 6 and 11. देखो यहाँ भी लिखा है, it's 6 and 11. These all are also 6 and 11. So warts are by 6 and 11. But malignant one, 16, 18, 31, 33, 45. This is the malignant. Say yes. Got it, everyone? So we are done with HPV. Read the question, tell me the answer. Read the question, tell me the answer. Which HPV causes condyloma acuminata? I'm asking condyloma acuminata. What is condyloma acuminata? You will see, ma'am, genital. Genital क्या? Cancer की wart. Benign को कहते हैं. और benign कौन सा कराता है? 6 and 11. So it's a very common question. Very repeatedly asked PYQ. Yes, है ना? So it's 6 and 11. Most common serotype causing cancer. Cervix cancer. Most common. Is it 6, 11, 16 or 33? I'm asking most common serotype causing cancer. Yes, so answer will be 16. It will be 16. You are right. The correct answer is 16. 33 also causes. The sequence is 16, 18, 31, 33 and 45. If 16 and 33 both are in option, I will go with 16. If 16 was not there, I will go with 33. समझ में आई क्या बात? So till now we have done 4 DNA viruses. Do you have any confusion till now? Do you have any confusion? Till now we have completed 4 DNA viruses. Which 4? I taught you pox virus. I taught you parvo. I taught you pox, parvo, adeno. And right now I taught you pepova. That is human papilloma virus, HPV. In the pox I taught you 3 viruses. Variola, vaccinia, molluscum contagiosum. Here we have only one member. Parvovirus B19. एक ही है ना? एडिनो में 52 टाइप्स आर देयर. हमने टेबल देखा. और यहाँ पे HPV is of 100 types, low risk, high risk. The low risk is 6 and 11 and high risk is 16, 18, 31, 33. Likewise. Do you have any confusion till now? Do you have any confusion till now? Tell me Mann syndrome. Mann syndrome यहाँ कहाँ से आया? Treeman syndrome. I didn't got. What syndrome? Tirupati, which syndrome you are asking exactly? Treeman. I've not heard this name. Is it tree and man? Treeman. Can you tell me the correct spelling? What exactly you are asking? I have not got. Is it related to virology? Or it is a random you are asking something? I didn't got it. Please elaborate your question. I will solve. If I know, I will solve it. Okay? Anyways. So we have done the 4 viruses till now. Can I start the 5th one? Herpes. Herpes is a very big virus. Very big virus. Among all the viruses, this is the biggest one. This one is the biggest. So I am teaching you herpes. Inside the herpes, we have to study 8 viruses. Herpes is of 8 type. Herpes is of 8 type. So can I start the herpes? That is the 8 type of herpes. I will teach you all. Last 3 are not very important, 6, 7, 8, but first 5 are ultra important. ultra important, herpes virus. Okay, so let's start herpes virus. Can we take a 10-minute break? Just 10-minute break. Hmm? I will have a cup of coffee. Hana, because my throat is, I'm having URTI right now. So the bad combination, my classes and my infection. So Hana, I will have some tea, green tea or coffee. So lubricate it, I mean, and I will be back. Hana, just in 10 minutes. And it's a big virus. So I do not want to leave it between. So it will take 45 minutes to complete herpes. And herpes के बाद अगला भी बहुत बड़ा है, hepadna, the hepatitis virus, another 1 hour. So 2 hours I require only for the next two viruses. Then DNA will be done, and finally we will come on RNA after that. है ना? So next two viruses are very big. ठीक है? So you two have a cup of coffee, tea, whatever you want, and we will start just in 10 minutes. है ना? So I will start at not 3:15. Okay, G. At 3:15, I will be back. And I'm not ending the class. Just switching off my mic and camera. Will it work? Just switching off my mic and camera. Okay, G. So I will be back just in 10 minutes. Hello everyone. Am I live? Am I back? Can we continue? Just a second. Can we continue? Someone please write down. So we were studying the viruses. We are studying the DNA viruses. We have completed 4 of them. I'm starting the 5th one. Okay. The 5th one is very big virus. I mean, there are 8 viruses in this family. So the next family I'm going to teach you is the herpes virus. So let's start with herpes virus. There are eight members here. You have to learn according to the numbers. Herpes virus 1, 2, 3, 4, 5, 6, 7, 8. You have to learn the names of each of them. Herpes virus 1 is known as HSV1. HSV1, what is the full form? Herpes simplex virus 1. Herpes virus 2 is known as HSV2. Herpes simplex virus 2. So 1 and 2 is very simple to learn. HSV1, HSV2. There is no problem in that. But you have to learn HSV3. HSV3 का नाम है varicella zoster. ये chicken pox कराएगा. It will cause chicken pox. ये chicken pox वाला virus है. Remember varicella zoster virus. We have seen chicken pox or smallpox के differences. Herpes virus 4 is known as Epstein-Barr virus, EBV. Herpes virus 5 is known as cytomegalovirus. ठीक है. 6 is known as H. इसको T बना लो. By mistake it is B. It is HT. नहीं. अच्छा. Okay. It's human B cell lymphotropic virus. Yes, it's the same. Human B cell lymphotropic virus. Herpes 7 is RK virus and 8 is Kaposi. ठीक है. So please learn the name. We will say it once. I'm teaching you herpes family. Herpes is a family. It consists of 8 members. 1, 2, 3, 4, 5, 6, 7, 8. One by one, you tell the name. 1 is known as HSV1. 2 is known as HSV2. 1 and 2 mein there is no problem. 3 is known as varicella zoster. 4 is known as Epstein-Barr virus. And 5 is known as cytomegalo. You have to learn these 5 in detail. I will give you the detail one by one. ठीक है. 6 is known as, what is the name of the 6th? The 6th is known as HBLV, human B cell lymphotropic virus. 7 is known as RK virus. And 8 is known as Kaposi sarcoma associated virus. Can I start? Are you people with me? Can we go ahead? So I'm teaching you herpes ka family. So let's start HSV1 and 2 together. ये दोनों मैं parallel लूंगी together. HSV1 and HSV2. Now remember a rule. HSV1 and HSV2, both of them cause the rash. It causes the lesions. Have you seen the lesions in the smallpox, chicken pox, molluscum contagiosum? In the same way, they also cause the rash. So the rule is that this is the waistline. हिंदी में कहूँ तो कमर, है ना? कमर के ऊपर, कमर के नीचे. So waistline. This is known as waistline. So above the waistline, at any part of the body, if the rash is there, especially on the face and the oral mucosa, it is caused by HSV1. है ना? And below the waist, especially over the genital areas, if the rash is there, it is caused by HSV2. है ना? So my point is that HSV1 causes rash above the waist and HSV2 causes rash below the waist. Learn this golden rule. Whenever they enter in the body, they cause the rash. Both of them cause rash. HSV1, HSV2. The rash look exactly similar. I will show you its vesicular rash. I will show the diagram. But the point is that location is different. HSV1 causes rash above the waist. HSV2 causes rash below the waist. The route via which they enter is also different. HSV2 enters via sexual route. It enters human body via sexual route and HSV1 enters via droplet. It enters via droplet, either droplet through inhalation or through direct contact. Through direct contact, है ना? Droplet or direct contact. But HSV2 enters via sexual. So please learn HSV1, HSV2. I'm teaching the first 2 together. Then I will teach you 3rd individually, 4th individually, 5th individually. So I have to teach you 5 out of the 8. I have to give you a detail of 5. है ना? So in the 5, 1 and 2 I am taking together. Please, है ना? So HSV1, HSV2. So write down or learn it. HSV1 spread via droplet or direct. HSV2 spread via sexual route. Anna, we have said that. Now, the point is that, let's say HSV1 or 2, anyone. I will take both. For example, this is the diagram, है ना? If HSV1 is entering via inhalation route, है ना, और HSV2 is entering via sexual route. चलो, HSV1 or 2, whatever is entering in human body, this is the baseline. Let's draw the baseline, है ना? They cause the lesion, है ना? For example, HSV1 most commonly cause lesion in the oral mucosa, buccal mucosa. HSV2 most commonly cause lesion in the genital area. ठीक है, lesions are exactly same, but the location is different. Listen my point. This rash is known as primary rash. What it is known as primary. So this is also primary rash above the waistline. Usually over the buccal mucosa, but anywhere on the body, but above waistline. है ना? This is usually over the genital, but anywhere below the waistline. Anyways, this is primary. This is primary. The rash is primary. It's painful. Multiple fluid-filled vesicles are there. I will show the diagram. After that, this virus, HSV1 and HSV2, both of them, they go in the nerves. They goes in the ganglion of the nerve. Let me draw ganglion here. Let me draw the ganglion of a nerve here. So the virus will go inside the ganglion of the nerve and remain dormant there for many years, many times. So it is known as latency. Both of them show the latency. Both of them show the latency. है ना? HSV1 become dormant in trigeminal ganglion and HSV2 remain dormant in the sacral ganglion. In the sacral. So they remain dormant for years. Whenever immunity drops, they again become activated, and every time they cause the same rush. I know they cause the same rush. So I mean it's recurrence. It's not one time. It will be recurrent many times. I know. So herpes occurs again. It is not one time. People take the treatment also. That time it will be cured. That time it will be subsided. But the virus is present in the ganglion for years, and it gets reactivated every time. Say yes if you got it. So the rash is repeated. Every time HSV1 will be above the waist. Every time HSV2 will be below the waist. People respond, say, you have any doubt? So that is the pathogenesis, latency. The most important feature is latency, which is seen in both 1 and 2. HSV1, HSV2, both of them show the latency. Got my point? You can see the same here also. Let me explain the meaning of this diagram. Can you see here the virus is entering and producing the lesion? These are the lesions on the skin. These are the lesions, and above the waist or below the waist. After that, look at the arrows. Look at these arrows. The virus is going in the nerve, resting in the ganglion for years, and again get reactivated. Look at the arrows coming back and producing the rash again. So doing rash, going, resting, coming back, doing the rash again. So this is the thing. So point is that HSV1 causes usually oral lesion, which are above the waist. Not only oral, above the waist, any lesion, and they rest in trigeminal ganglion. HSV2 causes lesion below the waist, but usually it's genital, and it is resting in the sacral ganglion. And after that, whenever they get reactivated from their ganglion, every time this causes this causes this. So repeated episodes of rash are there. My point is that. Now see, I will show you the diagram of all 5, but have a look. These are the types of the rash. Hannah, you tell me which is caused by HSV1, which is caused by 2. You tell me orofacial. Obviously it is 1. Obviously. You tell me nervous system. Obviously it's 1. It's above the waist. Now, cutaneous can be both. 1 and 2. Above waist, below waist. Ocular, again 1. It's above the waist. Hannah, genital, it's below the waist. So it will be 2. Hannah, visceral and disseminated is also 2. Hannah, multiple viscera in the abdomen, it's below. It's 2. Hannah, and herpes, neonatal herpes kaise hoga? What is the root of neonatal herpes? The neonatal herpes is through placenta. It will be transmitted through placenta. So placenta kahan aata hai? Above the waist or below the waist? The uterus and the placenta. Obviously below the waist. So it is also by 2. Got my point? So some are 1, some are 2. No need to learn. Learn the golden rule. Above waist, below waist. You can do that. Okay. See, one by one I will show you all. Let's start with orofacial. As I told you, orofacial is caused by 1. Above the waist. It is the most common manifestation of the 1. Most common manifestation of the 1 is orofacial. Can you see the rash? The rash is in the orofacial. Orofacial buccal mucosa. In the buccal mucosa. Sometimes the rash is on the lips. This lips. It is known as herpes labialis. Herpes labialis. Labialis is lips. Herpes labialis. It is known as herpes labialis. This is the image-based question in FMG. Herpes labialis. Small vesicle. If you rupture it, to rupture any of the lesion, the fluid will come out. This is known as vesicle. Vesicles contain fluid. Vesicle, है ना? Vesicles contain fluid. ठीक है? This is a vesicle. Coming on, cutaneous can be both above the waist, below the waist. HSV1, HSV2, है ना? The next you can see in cutaneous there are some special form. We all are doctors. So just suppose I'm managing, I'm doing a procedure on some patient. The patient have the herpes and I got a cut, and with the same scalpel with which I'm operating the patient, or I got a needle stick injury. So basically with the same herpes, it get, my finger get exposed, so I can have lesion on the fingers, है ना? So it is known as herpetic whitlow. In herpetic whitlow, the lesions are present on the finger, especially dentist में होता है. The dentists, they are operating or doing some procedure inside the patient mouth who's herpes positive, है ना? Hospital personnel में ज़्यादा होता है herpetic whitlow. So this is the image of herpetic whitlow. Have you heard about the wrestler? Who is a wrestler? Who do the punching, wrestling, है ना? In the wrestlers, the lip injury is very common because of the punch. Because of the punch, the lip injury is common. So they have lesion over the lips, the big lesions over the lip. These are known as herpetic glanditorium. So please learn the two terms: herpetus whitlow and herpetus glanditorium. Okay, what you are asking, Elizabeth? Which spread? Spread means exactly. Please elaborate the question. Spread means what you are asking. ठीक है? You see the genital, see male, see female, see the penis, see the vulva, and in both of them the lesions are like that. The lesions are multiple, tiny, 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 multiple fluid-filled vesicle lesions are there on the genitals. They are very painful. So say they are bilateral, they are painful, they are multiple, they are tiny, they are vesicular. Vesicular means they contain the fluid inside that. It is caused by two, not by one. It is caused by two, not by one. ठीक है? If the vesicles on the lip break, if it will break, fluid will come out, and secondary bacterial infection can occur and pus will fill. Okay, so they can break. There is no point in that. I mean, they can break. It can spread to other areas where it is spreading. I know. Anyways, so we are done with one. We are done with two. I know, one and two. Coming on three. What is three? What is the name of the three? I'm asking. What is the name of the three? Herpes three. Varicella zoster. Listen, my point. Listen, very important. Listen. I'm teaching you varicella zoster. I'm teaching you varicella zoster. Actually, it is a one virus. Hana? This virus have two components. As soon as this varicella zoster enter in human body, just suppose it is entering in my body. So the varicella component of the virus, there are two components, varicella and zoster. Virus is one, लेकिन components दो हैं, है ना? Two half हैं. So varicella component will cause the chicken pox. It will cause chicken pox. It will cause chicken pox in which the rash occur to entire body. Do you have chicken pox in your childhood? The rash is bilateral. Entire body, except palm and soles, everywhere rash is there. Hana, that is chicken pox. It's the primary infection. First time it occurs. Then what happens after that? After that, the virus rest become latent in the body, like HSV1, HSV2 in the ganglion. In the trigeminal ganglion, it rests for years, years and years. After that, in old age, when the immunity drop, at that time, zoster component become activated, not varicella, zoster. And it causes another disease known as shingles. It is known as shingles. Here also rash occur, but the rash is unilateral. The rash is unilateral and it is always on reactivation. Yes, it is always on reactivation. Got it? I will show you the images. To me or to many people, chicken pox already occurred. In future, we don't know we have shingles or not. Maybe in future I can have shingle. Maybe the virus is latent in my ganglion. We don't know. Shingle usually occurs after 40s or old age. Okay? So I'm not that way old, but yeah, in future it can occur. Okay? So chicken pox and shingles, the two diseases I told you here. So let's move varicella zoster. Varicella zoster. See, I'm showing you a diagram. So first time whenever the virus is entering, it will cause chicken pox. This is caused by varicella. And later on, the shingles, it is known as zoster. It is caused by zoster. And in between the two episodes, the virus is latent. The virus is latent. I'm repeating, Harsh. I'll show you a diagram and then I will repeat. Can you see? Varicella zoster virus is one virus. Varicella zoster virus is one virus, but it is having two components. What are the two components? One component is varicella, one component is zoster. So as soon as the virus enter, first varicella activate and causes the disease. Zoster don't cause the disease. So varicella cause the disease when the virus enter. For example, the virus enter via inhalation route. So it is entering in me. So via inhalation, as soon as it go in my body, the varicella component will cause the rash bilateral. bilateral rash, and the disease is known as chickenpox. This is chickenpox. After that, the virus go in my ganglion and it will rest for years, years, years. After that, in old age, the virus get activated again and come again. On reactivation, it come again. This time also it will cause rash, but this time rash is unilateral. By unilateral, I mean either right half or left half. The rash never cross the midline. If it is right half, it will remain right half. If it is left half, it will remain left half. It will not cross the midline. It is a peculiarity of the shingles. So zoster causes another disease which is known as shingles. Shingles may rash is unilateral. So chickenpox may rash is bilateral, shingles may rash is unilateral. Please learn that. Chickenpox is caused by varicella. Shingles is caused by zoster. है ना? This is the primary disease which occurs on first exposure, and this is a disease of reactivation which occurs later on. You got it? Harsh, you got it? Others, you got it? See, this is chickenpox. Can you appreciate chickenpox? This is the sternum. है ना? So rash is both side. You can see sternum के दोनों side. Sternum नहीं, backbone. Vertebra के दोनों type side है. You can see both side rash. But here you can see vertebra. है ना? You can see it's unilateral. It's unilateral. How many of you got it? How many of you got it? So I told you varicella zoster virus. It is having two components, varicella and zoster, the two components of the virus. Varicella causes chickenpox and zoster causes shingles. I know this is bilateral, this is unilateral. This is not very painful. Here nerves are also involved. It's very painful. I know this is primary. This is reactivation. Reactivation. ठीक है? How many chances of repeating chickenpox again, or it's impossible? Who's this? Lalit. Chickenpox will never occur again. Chickenpox occurs only once in life, but after that shingles can occur. I know, because for chickenpox, once if you have chickenpox in your life, the antibodies are present in the body. I have the chickenpox in my childhood, so my blood have the antibodies for the chickenpox, and they are very protective. 90% is the protectivity rate. So if again the virus entering in my body, chickenpox will not occur. Antibodies are already there, but maybe the virus is latent in me. Hannah, I don't know. And in future, I can have the shingles, but chickenpox will never repeat. Got my point? So that is the thing. Anyways, can we proceed, Hannah? So chickenpox and shingles. Chickenpox caused by varicella, shingles caused by zoster. This is primary infection, this is reactivation. This is bilateral, this is unilateral. This occurs in children, this occurs in adults. Any confusion? Anyone have any confusion? Respond, people. Smile and respond. Can we go ahead? Huh? Okay, so let me tell you the complications of both here. So varicella, zoster, ठीक है? Chickenpox, shingles, ठीक है? This I already told you. This is bilateral, unilateral. I already told you. Now let me tell you the complications of both. Hannah, so what are the complications of chickenpox and what are the complications of shingles? ठीक है? Can you tell me the complication? Chickenpox से क्या complication है? Chickenpox किस में होता है? I told you this one usually occurs in children and this one usually occurs in adults or old age. Hannah. So chickenpox occurs in children. What does children do? They just itch. Hannah. The vesicles will rupture, the fluid will come out, and they will have secondary bacterial infection. I still remember when I was a kid, I was having the chickenpox. I used to itch. It's very itchy. Hannah, it's painful and itchy. So my mother used to scold me, don't itch, just do a gentle massage like this, like this. So now I understood why she was saying that. Hannah. So if you itch, if the children are itching, so secondary bacterial infection because of the rupture of the vesicle will occur. Secondary bacterial infection in the skin. That is the most common complication. Most common complication is this. But what if the child is not itching and doing gentle massage? So this complication is not there. So extracutaneous complication. I'm sorry. The second most complication here is the meningitis. Meningitis. Yes, it's extracutaneous. Extracutaneous may it will be meningitis. Got my point? So that occurs in children. These are the two complications in children. Hannah, most common is skin. Second is meningitis and brain. Hannah, skin is due to itching. And extracutaneous is meningitis. What about adults? Adults में क्या complication है? Adults में it occurs pneumonia. If chickenpox is occurring in adults, so pneumonia is the complication. Pneumonia is the complication. Now coming on the complication of the zoster. Zoster may, after causing chickenpox, the virus go in the ganglion and remain latent there, and then it become reactivated. So when it become reactivated, so rash is there, which is unilateral, but nerve is also involved. The nerve is also involved. The nerve in which it was resting, वो nerve be involved होगी. Now which nerve? If it is an ophthalmic nerve, it will lead to blindness, है ना? Ophthalmic zoster. If it is a seventh now, it will lead to facial palsy. It will lead to facial palsy, which is known as Ramsay-Hunt syndrome. I will show you. Okay, so these are the complications. Firstly, the complication of chickenpox. In children, it is secondary bacterial infection due to itching, है ना? Or it can be meningitis in the brain. The extracutaneous, ये children में हुई. बड़ों में, adults में, it's pneumonia. Adults में it's pneumonia, है ना? And shingles, I told you, it's unilateral, है ना? Pain there, neuritic pain, vesicles are there. See the unilateral. Appreciate the unilateral rash. ठीक है? Complications. If the ophthalmic nerve is involved, ophthalmic zoster. If seventh nerve is involved, it is facial palsy. This one is facial palsy. It is known as Ramsay-Hunt syndrome. Facial palsy, Ramsay-Hunt syndrome. Okay. Yes, shingles occurs in adults, Bilal. Okay, got it? Got it? Any confusion? Do you have any confusion? So let's solve the question. Read the question, tell me the answer. Rash in chickenpox is what? What is correct for the rash in the chickenpox? What is correct? Can you please tell me the answer? Quick, fast. Can you please tell me the answer? Yes, rash in chickenpox. Is it deep-seated? Is it centripetal? It is monomorphic? It is umbilicated? I help you to learn CP for CP. कभी मत भूलना. CP for CP पे बहुत क्वेश्चन आता है. Chickenpox, centripetal. है ना? So this rash is centripetal. Who is deep-seated? No, chickenpox is superficial. Deep-seated smallpox होता है ना? Monomorphic भी smallpox होता है. यहाँ पे pleomorphic है. Umbilicated is molluscum contagiosum, not chickenpox. So B is the correct answer. Got it? Got it? Most common complication of chickenpox in adult. देखो question आराम से पढ़ना. Most common complication of chickenpox. I am asking in adult. I am asking in adult. Yes, I am asking in adult. So read the four options. Is it bacterial infection, meningitis, pneumonia, nephritis? Yes, Karthik, Megha, you all are right. To Bengal, everyone is right. I cannot read the names. Yes. So correct answer is pneumonia. In adults, I told you pneumonia. So let me change the question. From adults, let's do it children. Let's do it children. Now what is your answer? You should change your answer now. What is your answer now? Can you change the answer from the same four options? Most common complication in children. What does the children do? They just itch. They just itch. So secondary bacterial infection. In that case, the answer will become A. In that case, answer will become A. Not B. Not B. Someone is saying answer is B. Answer will be A. No? Most common complication in children is secondary bacterial infection. Yes, let me change the question again. Most common extracutaneous complication in children. Most common extracutaneous complication in children. Then the answer will become meningitis. You got the three questions, है ना? So most common complication. Most common complication, है ना? Most common complication in children. ठीक है? Most common extracutaneous complication in children and most common complication in adult. So three questions, three answer. So बच्चों में तो most common secondary bacterial infection है ना? Extracutaneous में meningitis है और adults में pneumonia है ना? So please learn. These are the questions which are already PYQ. So don't rush. In your exam, you have to look if they are asking children or they are asking adult. So means just give a minute to the question and then solve. So we are done with HSV1. We are done with HSV2. We are done with varicella zoster. So can I come on 4? Type 4. What is the name of type 4? What is the name of type 4? What is the name? It's Epstein-Barr virus, EBV. So can I start EBV? Let's start EBV. Epstein-Barr virus. Epstein-Barr virus aate se, I remember its other name. It's kissing virus. Yes, you heard it correct. Kissing. It's kissing virus. Why it is known as kissing virus? Because it is present in the saliva. It is present in saliva. It is transmitted from one person to another via kissing. Via kissing. That's why it is known as kissing virus. है ना? Epstein-Barr virus. Just learn its other name. You will remember everything of that. So just suppose, है ना? Just see. It is known as kissing virus. It is known as kissing virus. Epstein-Barr virus. The next one, the fourth, HSV4. है ना? It is. So just suppose it is transmitted from one person to another person via kissing. है ना? So it will there in the saliva. है ना? From the saliva, where does it go? Where does it go? From the saliva, it will go to the pharynx. है ना? It will go to the pharynx, nasopharynx. In the pharynx, we have tonsils. So basically, it will go on the tonsil. So Epstein-Barr virus will go on the tonsil. In the tonsil, we have B lymphocytes. Can you see? These are the B lymphocytes. So the Epstein-Barr virus का target है B lymphocyte present in the tonsil. So it will go to the tonsil It will go to the B lymphocyte. It will go to the B lymphocyte. This is B lymphocyte. This is the nucleus. This is the DNA. And this is Epstein-Barr virus. This is Epstein-Barr virus. And this is the B lymphocyte. Got it? So it will go inside the B lymphocyte with the help of a receptor. The receptors are known as CD21 receptor. It's a PYQ. With the help of CD21 receptor, it enters inside the B lymphocyte. After entering inside the B lymphocyte, it will form a protein. The name of the protein is LMP1 protein. Remember, HPV was forming E6 and E7. वो E6 और E7 बना रहा था, ये LMP1 बनाता है. So these are the questions from the pathology we are covering here. ये पैथो के क्वेश्चन है ना. LMP1 will go to the nucleus and do multiple mutation. Don't learn the name multiple mutation. Multiple mutation because of which it will have Burkitt lymphoma. यहाँ पे कौन सा लिम्फोमा होगा? Burkitt lymphoma, है ना. So you can see it's CD21. Epstein-Barr virus is entering in the B lymphocyte with the help of CD21, and after entering, it is forming LMP1 and causing many malignancies. One of them, most important, is Burkitt lymphoma. Burkitt lymphoma भी होगा, nasopharyngeal carcinoma भी होगा, Hodgkin's and non-Hodgkin's are rare. कभी-कभी हो सकते हैं. 4 malignancies: Burkitt lymphoma, nasopharyngeal carcinoma, Hodgkin and non-Hodgkin. है ना? Apart from that, it causes one benign disease also. The name of the benign disease is infectious mononucleosis. As I told you, HPV, first oncogenic virus बताया मैंने. EBV, second oncogenic. I will tell you 7 oncogenic viruses. The 2 are here. Anna, I taught you HPV also. I taught you EBV also. Anna, so HPV was causing having 2 protein E6 and E7, and this is also having a protein LMP1. Anna, this is also causing mutation. This is also causing mutation. This one also causing benign and malignant disease. This one also causes benign and malignant disease. Anna, so benign disease here is known as wart, and malignant disease is known as genital cancer, neoplasia or genital cancers. Yaad aaya? Here it is 6 and 11. Remembered? Here it is 16, 18, 31, 33, 45. ठीक है जी? Here benign is known as infectious mononucleosis. And the malignant, I told you 4 malignancies to learn. I told you 4 malignancies to learn. कौन सी 4 malignancies I told you to learn? Burkitt lymphoma, nasopharyngeal carcinoma, Hodgkin's lymphoma, non-Hodgkin's lymphoma. So you have to learn all this. And here malignancies में males में penis, females में cervix. So these are the oncogenic viruses. Till now I taught you 2. 5 more I will teach you today. Total 7 viruses are oncogenic. You have to learn their genes also. I mean proteins also, which is causing the mutation. To be specific, here E6 was causing mutation in P53 and E7 was causing mutation in retinoblastoma. Both of them are tumor suppressor genes. LMP1, don't learn the genes. They are complicated, but it causes mutation in multiple genes. You can learn like that. How many people are getting it? How many people are with me? Respond. Got it? Got it. So here benign disease is infectious mononucleosis. Infectious mononucleosis, also known as kissing disease, because it occurs via kissing. Also known as glandular disease. So here patient have simple tonsillitis, the pus in the tonsils. Patient have fever, malaise, and tonsillitis. That's it. And apart from that, the malignancies are there. Okay, tell me the diagnosis. How we do the diagnosis of infectious mononucleosis? I'm teaching you EBV. EBV causes benign disease and malignant disease. Kissing disease. EBV से मुझे एक ही word याद आता है, kissing. Kissing याद आया तो I remember the complete story कि it is in saliva. Kissing से कहाँ आएगा? Saliva में. From the saliva it will go to the pharynx. Pharynx it will go to the B lymphocyte. B lymphocyte से it will cause 1 benign disease in the tonsil and 2 malignant diseases. Benign कौन सी कराएगा? Tonsil में. See, this is benign. Infectious mononucleosis. और malignant भी यहीं कराएगा. It is nasopharyngeal carcinoma and Burkitt lymphoma. Here in the tonsil. So all the diseases are, sometimes it causes Hodgkin and non-Hodgkin also. So for infectious mononucleosis, what is the diagnosis? How you do the diagnosis of infectious mononucleosis? So there is a test known as Paul Bunett test. नाम सुना है? कहीं तो सुना है नाम? Paul Bunett test. So let me tell you what is Paul Bunett test. Shall I explain what is Paul Bunett test? Shall I explain what is Paul Bunett test? समझाऊं क्या होता है? Paul Bunett test is a heterophile antibody test. देखो. I will explain. Let me draw Epstein-Barr virus. ठीक है, ये Epstein-Barr virus है. ठीक है, let me draw sheep का RBC. sheep का RBC. आप बोलो मैम क्या कनेक्शन है? Epstein-Barr virus एक वायरस है, sheep के अंदर RBC, why you are drawing the two? The two are very separate things. The point is that the antigens present on Epstein-Barr virus, the similar antigens are present on sheep RBC. God has given the similar antigens. God has given the similar antigens. Yes, and this is known as heterophile. Heterophile, है ना? So we utilize this principle. We utilize this मतलब synonymy, and we utilize in diagnosis. How we utilize? So, for example, I'm having a patient in my clinic, a young 20, 25-year-old boy or girl having in my clinic, है ना? The boy or girl, they are having, I mean, the person is having tonsillitis, severe tonsillitis, and fever and malaise, and they are young. So the kissing history may be available. So I'm having high suspicion that the patient is having infectious mononucleosis. I know the tonsillitis is there and they are young and the history of kissing is available. Maybe the two person having it together, they are the partners or whatever. Okay? So that is infectious mononucleosis. I'm suspecting. How should I confirm? How should I confirm? What is the way I can confirm? So you will say, ma'am, if they have infectious mononucleosis, it means in their blood, Epstein-Barr virus is present. I agree. In their blood, Epstein-Barr virus is present. Hana, this is Epstein-Barr virus. I'm not testing Epstein-Barr virus. I do not want to test it. I want to test antibodies against the virus. Body may have formed antibodies against the antigen. No? I want to test that. So I will take a test tube and I will collect the serum. Serum contains antibody. Serum में antibody आती है. Serum में antigen नहीं आता. है ना? So I want to know if antibody is present, antigen is definitely present. है ना? If my blood have the antibody of Epstein-Barr virus, means I'm having the infection. Antibody cannot be formed de novo. Antibody will be formed after infection only. So if someone is having antibody, that person is having antigen also. That is my point. So this is the serum. है ना? So these antibodies are actually formed against the EBV. But what I will do, I will take a slide. On the slide, I will take one drop of sheep RBC. They are available commercially. Commercially मिलते हैं sheep के RBC. Sheep RBC, one drop. And one drop of your specimen. You put one drop of your specimen. है ना? As soon as you put the drop of specimen, now there are two possibilities. Specimen have antibody or do not have antibody. वही तो पता करना है. If the specimen have antibody, the antibody will bind with the sheep RBC. Why? Why they bind with the sheep RBC? You will say, ma'am, antibodies are formed against EBV. Why they are binding with sheep RBC? It is not EBV. But the antigen on the EBV and sheep RBC are same. Cross reactivity, heterophile. समझ में आया क्या? So you can see agglutinate dot granularity. How many of you got it? How many of you got the principle? So basically, from the patient, collect the serum. Okay, Elizabeth? Okay, everyone? Collect the serum. Collect the serum from the patient. Okay, G? Collect the serum from the patient. Just suppose there is a patient in front of me and I am suspecting infectious mononucleosis. I will collect the specimen. This is my test tube. I have collected the specimen. This is serum. I will take a slide. I will put one drop of sheep RBC and one drop of the patient's serum. I will mix with the stirrer. If dot granularity is there, it's positive. Not there, it's negative. It's known as Paul Bunnet test. It is known as heterophile. Heterophile का मतलब क्या है? What do you mean by heterophile? Two different species. One is a virus, Epstein-Barr virus, and one is a sheep RBC. Sheep, यानी भेड़, sheep का RBC. The antigen on both of them is same. This is antigen and this is antigen. So two different species have same antigen is known as heterophile. Heterophile में बहुत question आता है. You got my point? So this is the principle of the Paul Bunnet test. Nobody will explain you with such a clarity. See, this is the patient's serum. ये patient का serum है, having the antibody. And this is the sheep RBC. Once you mix the two on the slide, you will get the agglutinate. You will get the dot. Okay, G. Paul Bunnet test. Read the question, tell me the answer. We are done with 4 also. 1 हो गया, 2 हो गया, 3 हो गया, 4 हो गया. Now 5. HSV, herpes 5. That is cytomegalovirus. We have to see the 5th one, the last one. Okay, read the question. Tell me the answer. Epstein-Barr virus action on the nasopharynx is through which receptor? Via which receptor it enters in the nasopharynx B cell? Which receptors? Via which receptor it enters in the nasopharynx? It's CD21. Yes, via which receptor? Absolutely right. The correct answer is CD21. Via CD21, it enters inside. Next question, Paul Bunnet test. It is for which disease? Malta fever, typhus fever, enteric fever, or infectious mononucleosis? What is the correct answer? What is the correct answer? Yes, obviously, Paul Bunnet test is for infectious mononucleosis. And it's the heterophile test. You know what is heterophile? Heterophile agglutination test. Okay, coming on the 5th one. So till now we have seen HSV1. We have completed HSV2. We have completed varicella zoster. We have completed Epstein-Barr virus. And now I'm coming on the fifth one, cytomegalo, the last one. The other three are not important. Total 8 are there. है ना? So coming on the last one, technically last one, cytomegalovirus. देखो megalo में मुझे दिखता है mega, mega. You know mega cell, mega. Mega मतलब क्या होता है? Mega मतलब big, बड़ा. Why it is known as mega? Because among the 8 members, this is the largest. The fifth one is the largest. Cytomegalovirus is the largest among all 8. That's why known as mega. Mega matlab big. Number one. Number two, there is one more reason. So in whatever host cell the cytomegalovirus enters, after entering cytomegalovirus, it increases the size of the cell. It increases the size of the host cell. That's why also it is known as mega. So there are two reasons to say it mega. Number one, it is the largest virus in the family among the 8. Number two, it causes enlargement of the infected host cell. दोनों वजह से इसे कहते हैं मेगा वायरस, मेगा cytomegalo. Now, what is the clinical presentation? What does it do? So the clinical presentation varies in the 3 different individuals. What will happen if it is spread congenitally from a pregnant lady to the fetus? What will happen to the fetus? Number one. Number two, what will happen in the immunocompetent adults like me, you, who are immunocompetent, who don't have any immunosuppressed disease like AIDS or any other disease? And what will happen in the immunocompromised people? So one by one, we will see the three scenario. The first is the congenital. Congenital is via placenta. If the mother is having CMV infection during pregnancy, the fetus will have it. Multiple things occurs in fetus, लेकिन most important जो है brain छोटा रह जाएगा. The brain will be small. Such a fetus have a small brain. Microencephaly is the hallmark. Microencephaly. In the brain, multiple calcifications are there. Cerebral calcifications. So these are the hallmark. Small brain with calcification. Learn. Small brain with calcification. I'm teaching you CMV. Congenital CMV infection. Say yes. Hannah? Okay. Got it? Yes. The baby is born with CMV. It is microencephaly. Yes. Absolutely right. Hannah? So it is the most common intrauterine infection associated with congenital defect. Hannah? The baby have petechiae, hepatomegaly, splenomegaly, jaundice, small brain, calcification, prematurity. Hannah? And it occurs in early pregnancy. है ना, अर्लियर इज द प्रेगनेंसी, आई मीन फर्स्ट ट्राइमेस्टर में हुआ तो सबसे ज्यादा इफेक्ट होगा. सेकेंड और थर्ड ट्राइमेस्टर में इट इज नॉट मच अफेक्टेड, द फीटस इज नॉट अफेक्टेड, बट ड्यूरिंग फर्स्ट ट्राइमेस्टर बिकॉज ऑर्गेनोजेनेसिस अकर्स इन फर्स्ट ट्राइमेस्टर सबसे ज्यादा, है ना. मतलब सेकेंड थर्ड में भी होगा बट हियर मैक्सिमम इन्फेक्शन चांसेस व्हेन इट अकर्स इन द फर्स्ट ट्राइमेस्टर. सो प्लीज लर्न फर्स्ट ट्राइमेस्टर, है ना. सो दैट इज द थिंग. इम्यूनोकॉम्पिटेंट में ये तो कंजेनाइटल हुआ, कंजेनाइटल इज डन. कंजेनाइटल में माइक्रोइनकेफेली, microencephaly and calcifications are important, है ना. Immunocompetent healthy adults में it will cause infectious mononucleosis like syndrome, not infectious mononucleosis but simple tonsillitis like that, कुछ खास नहीं कराएगा, simple tonsillitis like infectious mononucleosis. And last one, immunocompromised people. Immunocompromised people, if in the AIDS patient when the CD4 count is less than 50, in the AIDS patient it causes chorioretinitis. What does it cause? It causes chorioretinitis. And in organ transplant patient, I'm coming on it. Bilal, wait, listen. In AIDS patient, it causes chorioretinitis. You know what are organ transplant patient? The organ transplant patient are the patients in which we have done the transplant, and post-transplant we are giving immunosuppressive drug. After the transplant, we give the immunosuppressive drug. Now, so in them, within first 4 months of the surgery only, this is the most common viral infection patient may have, and the patient have pneumonia. You got my point? Imagine there is a person whose both kidneys are failed, both the kidneys. Now there is a donor available. Hannah, so the donor has given the kidney. This is donor. So you are giving this kidney to this patient. You have implanted, Hannah, but this is foreign. This is foreign. So the immune system of this person can reject the kidney because it is foreign. It is foreign for this body. We do not want that. We do not want that. So immediately after the transplant, we will give the person immunosuppressive drugs. We will suppress the immunity so that the graft is not rejected. But it is a double-edged sword. एक तरफ से the graft is not rejected, I agree, but person is at high risk of infection. So among those infection, among the viruses, most common virus after surgery is cytomegalovirus. How many of you got it? So it is the most common virus post-transplant. Post-transplant, most common viral infection within first 4 months, and it causes pneumonia. It causes pneumonia. Yes, Bilal, of course, it causes low birth weight. Prematurity and low birth weight can be occur in cytomegalovirus infection. Okay, can we go ahead? Diagnosis you all know. Owl eye appearance. You know or you don't know? Cytomegalovirus. Cytomegalovirus is owl eye appearance, है ना? Double inclusion bodies in the nucleus and the cytoplasm. Owl eye appearance. That is the diagnosis. Read the question, tell me the answer. Quick. Read the question, tell me the answer. Quick, people, quick. चलो, बताओ मुझे answer. What is the answer? Just a second. Yes. The correct answer is CMV. You all are right. Owl eye appearances, seen in CMV. You get that. One more question, take the clues. It's a clinical question. We will take the clues. There is a 52-year-old man undergo lung transplant. Okay? 52-year-old man undergoing lung transplant. 2 months later, the transplant, he developed pulmonary symptoms and he developed pneumonitis. 2 months after the transplant. 2 months after the transplant. Most common virus responsible. Most common virus responsible after transplant is cytomegalovirus only, causing the pneumonia. Pneumonitis. We are done. Hannah? So we are done with HSV 1, 2, 3, 4, 5, 6, 7, 8. Just learn the names. Not important. But 1, 2, 3, 4, 5 are done. Any confusion, any doubt? We are done with herpes. Right? Before going to the next, let me tell you something. Of course, whatever you are doing right now in your life, the phase, it's very hard phase. The hardest phase of your life. I agree. Anna, it's hard. But it was supposed to be hard now. Before entering in the medical field, you may be knowing that you are going to become doctor. You are studying to become doctor. I don't know whether you know it or it's the most difficult profession in this world considered. Yes or no? You are, responsible for the lives of the people. So it was supposed to be hard. And if it was that way easy, everybody would become do it now. But you are doing it, others are not doing it. It's a privilege. Utilize it wisely. Anyways, coming on the last DNA virus, finally the last DNA virus. There are 6 DNA viruses now. We are done with 5. Now coming on the last DNA virus, hepadina. Okay? I started with pox. I told you the 3 pox virus. Variola, vaccinia, molluscum contagiosum. I told you parvo, parvo में slap cheek appearance. याद आया? And then I told you adeno, space vehicle वाला. Papova में HPV I taught you. Herpes just now the 8 में से 5 herpes I taught you. Now finally I am coming on hepadina. How many of you with me? Can I start hepadina? Say yes. Okay. So let's start the hepadina virus. Again, it's a big virus. What is hepadina virus? Which virus come here? It is hepatitis B virus. In this family, only one virus come. The name of that virus is hepatitis B virus. Hana? You know how many type of hepatitis are there in our syllabus? There are 5 type of hepatitis. Hepatitis A, B, C, D, and E. Please understand. There are 5 type of hepatitis. है ना सबको पता होगा hepatitis 5 तरह का होता है. So hepatitis B is caused by a DNA virus. That is hepatinovirus. But the remaining 4 hepatitis A, C, D, E, these are caused by RNA virus. These all are caused by RNA viruses. Different RNA viruses. And I will tell you the names. Here the name is hepadina, the DNA का family. RNA में ये वाला picorna है. This one is picorna. This one is flavi, है ना? This one is delta, है ना? And this one is kelsey. So I told you the families na A, B, C, D, 4, TPH, tweet per hour. So they are coming from there. Got my point? Now the last DNA is left. I have taught you 5. The 6th one I am teaching you. What is my planning? Shall I tell you all hepatitis together? So उसमें होगा क्या? 1 DNA will be covered and 4 RNA will be covered. So anyways, after DNA, the next we have to start RNA now. So let's cover 4 of them together. So I am teaching you flavi also, kelsey. Kelsey कहाँ लिखा है? Kelsey also. I mean, I will teach you picorna, kelsey, and delta. Delta यहाँ लिखा नहीं है शायद, है ना? So delta. I will teach you delta also, है ना? So these 4 from here and 1 from here, total 5 together. So I'm teaching you hepatitis virus, the 5 type of hepatitis, A, B, C, D, E. There is one more hepatitis virus, G, but later on we found it don't cause hepatitis. Only 5 of them cause hepatitis. How many with me? Can I start? Hepatitis virus. Okay, G. One of them is DNA and the remaining all are RNA. So I'm teaching you 5 viruses together. जिसमें DNA का लास्ट हो जाएगा और RNA के फर्स्ट 4 हो जाएंगे. ठीक है, तो RNA में से भी 4 हो जाएंगे, एक DNA में से हो जाएगा. उसके बाद RNA में से 6 more I will teach you. तो RNA में से I taught you total 10. Out of the 15 I will teach you 10. 4 तो यही हो जाएंगे, उसके अलावा 5-6 और पढ़ाऊंगी अभी इन डिटेल, ठीक है। सो लेट्स स्टार्ट द हेपेटाइटिस वायरस, जस्ट अ सेकेंड। So let's start hepatitis. The 5 type of hepatitis virus A, B, C, D, E. Okay. First understand only B is DNA. First understand. Rest all are RNA. See RNA, 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 RNA. One more thing understand. All DNA are double stranded except RO. So this one is double stranded. And all RNA are single stranded except Rio. So यहाँ Rio तो है नहीं. So all of them are single stranded except B which is double stranded. कोई confusion इसमें? इसमें कोई confusion? Do you have any confusion? Learn the families of all 5. Huh? Learn the families. Here it's Picorna, here it's Hepadna, here it's Flavi, here it's Delta, and here it's Kelsey. Learned? Huh? So tell me. Now I'm telling you most important. A, B, C, D, E. The 5 type of hepatitis. I told you B is caused by a DNA virus. Rest all of them are caused by RNA virus. RNA virus, RNA virus, RNA virus, RNA virus. है ना? I told you the DNA are double stranded. They all are double stranded. And all the RNA are single stranded. So ये कहानी भी खत्म करो बोल के, है ना? उसके बाद tell me the family. So B की तो Hepadna. It's Hepadna. Remaining A की Picorna. A की Picorna. ठीक है? C की Flavi. D की Delta. And E की Kelsey. Please learn the families. Learn DNA, RNA. Learn single-stranded, double-stranded. Learn the name of the families. Do you have any confusion in that? Anyone have any confusion? Shall I proceed? इतना तो learn करो. You get many questions out of that. Learn the incubation period. Incubation period. Learn 30 days, 40 days, and center में 50 days. This is how I learned. 30, 40, center में 50. 2 बच गए. There are 2 left. Learn 60 to 90. 60 to 90 days. This is how I learned. So incubation period पे भी काफी क्वेश्चन आते हैं. Learn the incubation period. First learn DNA, RNA, then understand single stranded, double stranded, then understand the family of all 5, and then understand the incubation period. And now there is no rocket science in that. Just learn. It's very simple. And now you can learn like this. You can see it's 30, then 40, then central is 50, then 60 to 90. That is the incubation period. The same thing is written in front of you. Let me show you. Let me show you. 30, 40, 50, 60 to 90, 60 to 90. Got it? Any confusion? Now listen. Now listen. The most important thing. A, B, C, D, E. What is the route? How does it transmit from one person to another? A and E transmit by fecal-oral route. By fecal-oral route. Anna? And BCD, I will draw. BCD is transmitted not by fecal-oral route, either by sexual route or by parental route. Parental or by vertical. Let me explain. Hannah, please read. I will explain. I will explain. Hannah, listen. Imagine this is a person. This is a person. You can see. Hannah, this is another person. Okay? My diagrams are just rough to give you the concept. Okay? This person is having hepatitis A or hepatitis E. Okay? So basically A or E, both of them are shed in the feces. The virus is shed in the feces. A and E both. है ना? The feces will contaminate the food and water. And the contaminated food and water is ingested by another individual. And another individual also have A or E. So via this route, it is transmitted from one person to another. So this route is known as feco-oral route. This is known as feco-oral. This is the meaning of feco-oral. How many of you got it? How many of you got it? This is the meaning of feco-oral. I know this is the meaning of feco-oral, and it is seen only in A and E. Why only A and E? Because A and E are shed in the feces, not others. A and E are shed in feces, not others. Got it? Now one more thing. Listen, let me draw one individual. Let me draw one more individual. Now the first individual this time have hepatitis B or C or D, not A and E. BCD. BCD. I'm sorry. BCD. Either B or C or D. Point is that they are not shed in stool. They are not shed in stool like A and D. So there is no possibility of fecal-oral. No. Fecal-oral cannot be there because they are not shed in stool at all. So how does they spread from this person to this person? What are the ways? Sexual intercourse. The two persons are doing sexual intercourse with each other. This is how they can spread. Number one, but it's a rare route. More common, more common between them is parenteral. What do you mean by parenteral? है ना? You use a needle or a syringe in this and the same needle or syringe in this. So the same needle, same syringe, है ना? यहाँ पे blood product. It's very rare blood product भी. This blood is given to this, है ना? But the blood go via blood bank. Nobody in the India especially can give blood directly. Just suppose I need blood and you have it. So you cannot give it directly to me. You have to give to the blood bank. The blood bank will screen it and then release to the receiver. So it is via blood bank. But sometimes mistakes can occur in the blood bank. Via blood products can occur. So via needle, via syringe, via blood product, via organ transplant. I know it is transmitted from one person to another, and this is known as parenteral route. This is known as parenteral. And third is vertical. You can understand now what is vertical. This is present inside this individual. I mean, this is the baby. I mean, this is the newborn. I mean, this is the fetus, and this is the mother. So via placenta, it can transmit. Say yes. Via placenta, it can transmit. Say yes. Got it? So these are the three routes. What are the three routes? What are the three routes? I'm asking. What are the three routes here for BCD? Say, ma'am, parenteral, most common. उसके बाद sexual और उसके बाद vertical. है ना? Parenteral is common in doctors also. है ना? We are giving injection or doing surgery or procedure of some patient having hepatitis B, C, D. And the same needle, scalpel, cut or injury occurs to us. So in this way, it can get transmitted. So parenteral, sexual, vertical, the three routes. So same story is written in front of you. Everyone here, see? So A is by fecal, E is by fecal. There is no doubt in that. But what about BCD? In BCD, read the routes. Read the routes. Blood matlab parenteral. Parenteral route most common, sexual and vertical. Do you have any confusion? Can we proceed? So learn the route. Last thing you have to learn, please. The most important thing, last but most important, A, B, C, D, E. The 5 types of hepatitis. Listen, the 5 types of hepatitis. Listen. What are these I'm teaching you? ये पांचों क्या हैं? What is A, B, C, D, E? What is this? They are hepatitis virus. What do you mean by hepatitis? They all cause hepatitis? Yes, they all cause hepatitis. There are 3 types of hepatitis: acute, chronic, and fulminant. My next question to you: which of them causes acute hepatitis? Which of them causes chronic hepatitis? And which of them causes fulminant hepatitis? Can anyone tell me? Can anyone tell me? The answer is acute is caused by all. Acute is caused by all of them. All of them. Acute is very simple to treat. हमें acute से डर नहीं लगता, chronic से लगता है ना? Why? Acute is 99% curable, 99% recovery. No treatment even required. What are the symptoms? Patient have fever, है ना? Patient have loss of appetite, patient have jaundice. The liver is involved ना, तो jaundice हो जाएगा. Fever आएगा, loss of appetite होगा. Acute, 99% cure rate. But what about 1%? In 1% of them, acute is very severe. Pura, the complete liver undergo necrosis. It is known as fulminant. So 1% convert to fulminant, and that too occur in all 5. Because 1% of the acute is fulminant. And the death occurs, no treatment, in which complete liver undergo necrosis. That is only 1%, but it's very rare chance. 99% is recovered. What about chronic? What about chronic? Main is chronic. Chronic never occurs in A&D. No, chronic never occurs in A&D. Chronic occurs in BCD. BCD, chronic, chronic, chronic. Say yes. Chronic can occur in BCD, but not in A, not in E. Chronic is never cured. Never cured. There are 3 possibilities. It is never cured. Once occur, it will be there throughout the life. Acute 99% cure. Chronic never cured. Never cured. So हमें acute से डर नहीं लगता. Acute is not scary. Chronic is scary. हमें chronic से डर लगता है. Chronic में one possibility, the person will be carrier throughout the life. Or the person is having hepatocellular carcinoma and die. Cancer. Or the person will have cirrhosis and die. So these are also end diseases. Either carrier, hepatocellular carcinoma, or cirrhosis. This occurs in chronicity. Since there is no chronicity in A&E, so in A&E, no carrier, no hepatocellular carcinoma, no cirrhosis, no chronicity. But in BCD, these are the possibilities. How many of you got it? These are the basic concepts you have to learn. Say yes, people. Say yes. Say yes. Respond. I'm trying very hard to explain you the concept. I know. So that you have to understand. One more thing you have to understand. Listen, A and E don't have chronicity, no? A and E. A and E. They cause only acute. They don't cause chronic. तो दोनों को बुरा लग गया. They mind it. other friends are having the chronic hepatitis also, we cannot cause chronic, है ना, they are hurted. So don't worry, we said कि you are causing acute, ना, you are the most common cause of acute. You can't cause chronic, it's okay, but you are the most common cause of acute in children. A is most common cause of acute in children, and E is most common cause of acute in adults, है ना. So most common cause of acute we have seen. Say yes, got it. Can we go ahead? Okay. So this you have. With this concept, we can start the hepatitis. Hana? So let me see. I guess you can tell me all the things. Hana? So we will draw the table. Once you will tell me the answers, and then we will see the details, other details. Just a second. A. It's like this. First thing, you will tell me the DNA or RNA, I mean genetic material of all of them. Then you tell me single stranded or double stranded for each of them. Then you will tell me the family of each of them. Then you will tell me the incubation period of each of them. Then you will tell me the route of spread for each of them. Then you will tell me the acute, which one causes, fulminant, which one causes, chronic, which one causes. Anna, so that's it. Okay, yes, very good. Only B is DNA, rest all are RNA. Very good, Canel, very good, Elizabeth, very good. Rest all are RNA. I'm writing only R, it's RNA. ठीक है, चलो. Single or double stranded? So all RNA are single stranded. We know. है ना? Only Rio is not there. And all DNA are double stranded except parvo तो है नहीं. है ना? So that is single or double. Family की बात करो. चलो. Tell me the family. So here family is Picorna. है ना? B is Hepadina. है ना? C is Flavi. D for Delta. D for Delta. And E में Kelsey. Try to learn तो करना पड़ेगा. Eat walnuts and learn. ठीक है, चलो. Incubation period बताओ. How I learn incubation period? I explained you. Start with 30 days, 40 days, center में 50 days. 30, 40, 50. जो बच गए दोनों 60 to 90, 60 to 90, 60 to 90, 60 to 90. It's like this. है ना? So these are the incubation period. Okay, G. So genetic material is done. Single stranded, double stranded is done. Family is done. Incubation period is done. Now coming on root. So A and E ka root is pico-oral. Pico-oral. The remaining three ka root either parental, most common, or sexual, or vertical. I don't think you have any doubt in that. Please learn. Howsoever you learn, please learn. What about the acute? You will say, ma'am, acute is caused by all of them. So acute. 99% cured. 1% convert to fulminant. Okay, coming on the chronicity. Chronicity don't occur in A and E. Chronicity occur in BCD, है ना? And what is the fate of chronicity? The chronicity can lead to carrier state, it can lead to HCC, and it can lead to cirrhosis. Absolutely right. Super good, super. Not only this, A is causing only acute, not chronic. E is causing only acute and not chronic. So A is the most common cause of acute in children, and E is the most common cause of acute in adults. Got it? So you get many questions from this, believe me. Now I will give you detail of only two. You can read this table by your own. And the same thing is written. ठीक है ना? 3 more oncogenic viruses BCD. What I told you, देखो. A, B, C, D, E. I told you acute is caused by all, na? Acute is caused by all. Fulminant is also caused by all. But I told you chronic is caused by, chronic is caused by only BCD. Have I taught you? And what I told you, what is the fate of the chronic? The fate of the chronic is either HCC or cirrhosis. तो मतलब HCC का होता है ना इन तीनों में. BCD are causing the HCC. So these are the 3 oncogenic viruses. These are 3 more. So till now, how many oncogenic I told you? First oncogenic I told you EBV. No. First oncogenic I told you HPV. Second oncogenic I told you EBV. And now I am teaching you hepatitis B, hepatitis C, and hepatitis D virus. So 5 now, till now I have taught you. Hai This one was causing the cancers of the genital cancers. Hai Panis in male, cervix in female. And the benign one are known as warts. Warts and genital cancer. ठीक है? EBV में benign is known as infectious mononucleosis. और malignant में Burkitt lymphoma, nasopharyngeal carcinoma, Hodgkin's and non-Hodgkin's, है ना? And these three are causing HCC. I told you, today I will teach you seven viruses which cause cancer. 5 of them are done. Bacteria में only one bacteria. Bacteria में only one bacteria. That is H. pylori. Is what you are asking? Yes. Hepatitis E is the most common in pregnancy. You are right. Most common cause of fulminant hepatitis in pregnancy is E only. Yes. So I will teach you only 2 here in detail. Table में हमने सारे देख लिए. Most of the questions will be from table only. Now 2 detail I will give you, B and D. I will give you the detail of B and D. And now B and D are important. Let's start with B. B is difficult. Now, whether it's a warning or I'm scaring you, it's not like that. But B is difficult. Please concentrate. Please everyone see on the gazette. Can you see this is a diagram of hepatitis B virus? Can you see it's a double-stranded DNA virus? So you can see the DNA at the center. See the DNA at the center? It's a double-stranded DNA virus. Now see, there are 3 antigens on this virus. 3 antigens. Please understand. The first antigen is present on the surface. S for surface. Surface. है ना? It is known as hepatitis S antigen. S for surface. S for surface. The second is E. E for envelope. Envelope. It is present on the envelope. And third C. C for core. It is present inside the core. What are the 3 antigen? Can you tell the name? S, E, and C. Now imagine this present in blood of a human. This virus entered in the blood vessel of a human. Can you see a blood vessel? Inside the blood vessel it is present. So out of them, please mind my words, S will leak and come in the blood. So hepatitis S antigen leak and come in the blood. E also leak and come in the blood. Hepatitis E also leak and come in the blood, but C never come in the blood. No, C is present deep inside. It cannot leak. It will never come in the blood. So if you take the blood sample out, you can get 2 antigen, S and E. You will never get C. How many of you got this concept? C कभी blood में नहीं आता. हिंदी में बोलूं तो C never comes in the blood, है ना? S come and E come, but C never come in the blood. ठीक है? Now body will start forming antibodies against these. And now whenever this is present in the blood, body will form antibody against all 3. So see the antibody against all 3. Everyone. So body will form the antibodies against S and TS. You will say, Mama, okay, समझ में आया. Against, I'm sorry, against E and TE. Against E and TE. Very good, समझ में आया. अब C को लग गया बुरा, है ना? C is feeling bad. C is feeling hurted. My other 2 friends are leaking out. Why I am not leaking out? I cannot leak out because I am present in the core. So we said it's okay. You can't leak out. We will make your 2 antibodies. हम तुम्हारी 2 antibody बनाएंगे, है ना? So we have 2 anti-C. Anti-C are of 2 type, IgM and IgG. So total 4 antibodies are there. How many of you got it? Total 4 antibodies are there. How many of you got it? Total 4 antibodies. We are having total 4 antibodies. So we are having 2 antigen and 4 antibodies. What are the 2 antigen? First S leak and then E leak. है ना? Then body start forming antibodies. S के लिए we have anti-S. E के लिए we have anti-E. है ना? C don't come in blood. So we will make 2 anti-C. Anti-C is of 2 types, IgM and IgG. How many of you got it? So basically learn that IgM and IgG are the types of anti-C. They are the type of anti-C. So learn the sequence. In which sequence they come in the blood. Please understand the sequence. I'm teaching you the serological markers of hepatitis B. It's important. It's difficult. I'm teaching you the serological markers here. Listen, what is the sequence in which they arrive in the blood? Tell me the sequence. Obviously, first antigen will come and then antibody will come. So first antigen will come, then antibody. पहले antigen आएगा ना blood में, then body will form antibodies against that. So antigen में सबसे पहला antigen आएगा S. S सबसे पहले leak होके गया आएगा. The first antigen which come in the blood is S. The second antigen which come in the blood is E. S followed by E. The second antigen which come in the blood is E. First S will come followed by E. Now body start forming antibody. So body सबसे पहले किसकी antibody बनाएगी? हाँ? The body will form the first antibody against C क्योंकि C को बुरा लग गया कि मैं तो आ ही नहीं रहा. तो C की antibody is the first. There are 2 type of C की antibody. So first IgM will be formed. Then this is anti-C, IgM. Then anti-E, anti-E. Then again C, but this time it's IgG. And then S. The last antibody is S. So you get 4 questions here. Tell me the first antigen. Tell me the last antigen. Tell me the first antibody, the last antibody. First antigen is S, last antigen is E, first antibody is C, IgM-C, last antibody is S. Say yes, you got the 4 questions. So this is the sequence they come in the blood. Can you tell me the sequence again? एक बार फिर से बताओ. पहले antigen बताओ फिर antibody. What is the yes, yes, yes. Yes, Sukhchin. Yes. So what is the sequence of antigen? S followed by E. SE. Learn SE. What is the sequence of the antibody? What is the sequence of the antibody? Say CECS. Learn like that. Hana? The first C is IgM and the second C is IgG. You learn like that. CECS. It's like that. Got it? CECS. It will be like, so please learn. Whatever, howsoever you learn, learn. This is SE, this is CECS. These are antigens, these are antibody. This is the sequence. Got it? This is the sequence. What is S and E? These are the antigens channel. S and E are the antigens. Now, what exactly you are asking? S for surface and E for envelope. Can you see the 3 antigens channel? You see the diagram. On the diagram, can you see this antigen? This antigen is known as surface antigen. It is known as surface antigen. Surface. Surface ka hai yes, because it is present on surface. Or E for envelope. It is present on envelope. That's why known as E. So these are the antigen and C for core. C for core. So first antigen will leak. Na, ye antigen leak ho ke bahar aayenge. है ना, C cannot come. And then the body start forming antibody. Antibody तीनों के लिए बनेगी. Antibody will be formed for all of them. Got it? है ना? Now the sequence is done. है ना? Sequence is important for this graph. Actually, nobody is going to ask you the sequence in the exam. They will give you this graph and they can ask you the labeling of the graph. है ना? You cannot understand the labeling if you don't know the sequence. Now you tell me सबसे पहले कौन आता है? Who is the first to come? So this first, can you see this first graph? I'm marking with red color. Anna, let me change the colors. Just a second. Just a second. Can you see this first one? This one. This one. So obviously, what is it? You tell me. It is hepatitis S antigen. The first antigen which is coming. S ka antigen. Ideally, E ka alag dikhana chahiye, but in this diagram, E is also this. S and E both are this. Ideally, thoda sa gap hona chahiye. There should be 2 graphs. First S followed by E. But they are showing S and E together. ठीक है, कोई बात नहीं. Now start forming antibodies. You start forming antibodies. So the first antibody is which one? ये pink color का क्या है? I'm drawing with blue. ये क्या है? You will say, ma'am, it's anti-HBC. Anti-HBC. Can you see this is anti-HBC, which is IgM. Okay, the next. Can you see what is this one? This one. It is anti-HBE. उसके बाद क्या आता है? What is the next? Then again anti-HBC, but this time it will be IgG. है ना? और last में क्या आता है? Anti-HBS. है ना? IgG once come, never go back. HBS, anti-HBS once come, never go back. So these two never go back. They will be always ever. है ना? Always continue. So they can ask you the labeling of the graph. है ना? They can label any area and they can create multiple questions. How many of you got it? So this is how you have to make this graph. Can you make this graph? I mean, can you do the labeling if you know the sequence? Say yes. So you get image-based question on this graph very, very, very frequently. So you have to draw like this. है ना? You have to draw like this. The first antigen which is coming is HBS. It's like this. है ना? Let me label this as HBS ka antigen. The second antigen which is coming after little bit some time of this, it is HBE. है ना? This is HBE. Both are antigen. Now the antigens are over. Let's talk about the antibodies. The first antibody which is coming, is anti-HBC. Anti-HBC. And it's IgM. IgM wali. The next which is coming is anti-HBE. है ना? C के बाद आएगी anti-HBE. Anti-HBE. है ना? फिर again it will come. Anti-HBC, लेकिन this time IgG, it will never go. है ना? This is IgG. It will never go. And the last will come. Last will be anti-HBS. It will also not go. So you can do the labeling also. And if you know the sequence. So very frequently you get image-based question on this graph. People respond, say, समझ में आया? समझ में आया? Say yes if you got it. Now the last thing, which is very difficult portion of this. The last thing, very, very difficult portion here. Let me tell you the pairs. Let me tell you the relevance and their pairs. Listen. इनका मतलब क्या है? इनका relevance क्या है? What is the relevance of each of them? What is the relevance of each of them? Let me tell you. See the first pair. S and NTS. पहला जोड़ा देखो. S and NTS. Okay. I will come on the graph page again. Namrata, wait. Let me explain this. Then I will come. Okay. Listen. S and NTS, the pair. Please understand, it's very difficult. S and NTS never present together. One of them at a time. One of them at a time. Either S ka antigen or S ka antibody. They will never present together. If S ka antigen is there, it means the patient have the infection. Right now, the patient have the infection. If S ki antibody is there, means the patient is recovery. The patient is in recovery. So both cannot occur together. Either the person have the infection or the person have the recovery. Both cannot occur together. Got my point? Got my point? So S ka antigen indicate infection. S ki antibody indicate recovery. Recovery or vaccine. Are you vaccinated for hepatitis B? If not, get it today only. All healthcare providers, all the doctors should be vaccinated for hepatitis B. So if you are vaccinated in your blood, I am vaccinated. In my blood, anti-HBs is present. In your blood also, anti-HBs is present if you are vaccinated. है ना, recovery or vaccine, the same thing, है ना. So that is the first pair. Coming on the second pair. Second pair is E ka antigen or E ki antibody. Again, they are never present together. One at a time. One at a time. Either E ka antigen not antibody or E ki antibody not antigen. What does they indicate? What does they reflect? What does they indicate? So, and they indicate infectivity. E का एंटीजन है तो मतलब हाई इन्फेक्टिविटी. इट मीन्स द पर्सन इज हाईली इन्फेक्टिव, हाई इन्फेक्टिविटी. एंड ई की एंटीबॉडी है मतलब लो इन्फेक्टिविटी. इन्फेक्टिविटी इज द टेंडेंसी टू इन्फेक्ट अदर्स. इन्फेक्शन नहीं है ये, इन्फेक्टिविटी है. फॉर एग्जाम्पल ई का एंटीजन है, सो इफ दिस पर्सन, फॉर एग्जाम्पल I am giving an injection to a patient. The patient is in front of me. Anna, I have given the injection. I was taking out the needle. I was discarding it, but by mistake, I got a needle prick injury. I got scared. Oh my God. I have checked the history of the patient. Oh my God, the patient was hepatitis B positive. The patient is HBV positive. He's a known case of HBV positive on treatment. And investigation, while doing some investigation or while giving some treatment, I got a needle prick. I got scared. मुझे भी हो जाएगा hepatitis B. I did not want that. So I will check the hepatitis E antigen and antibody status. If the patient के blood में hepatitis E का antigen is present, there are high chances कि मुझे होगा, मुझे भी हो जाएगा, because infectivity is very high. And if antibody is present, there are very less chance कि I will get the infection. You got the meaning? You got the meaning? Infectivity. Infectivity is the tendency to infect other. That is infectivity. Communicability. You can say the word communicability. People say, got it. हाँ ना तो बोलो बेटा. Say something. Got it. है ना? So okay. So that is the meaning of the infectivity. है ना? Coming on the last pair. We have seen the pair of the S. We have seen the pair of the E. Now we are coming on C. C का antigen तो आता ही नहीं है blood में. C antigen never come on the blood. है ना? The two antibodies are there. M or G. Now, S ka antigen was indicating infection, but whether the infection is acute or chronic, we don't know. We don't know. It can be both, है ना? So M मतलब acute infection or G मतलब chronic infection. How many of you got the 6 relevance? I'm telling you what does they indicate. So I taught you hepatitis S का antigen और hepatitis S की antibody. Antibody के आगे we write antibody, है ना? Then I told you hepatitis E का antigen और हेपेटाइटिस ई की एंटीबॉडी. एंटीबॉडी इज एंटी, है ना? एंड लास्ट आई टोल्ड यू ओनली 2 एंटीबॉडीज. हेपेटाइटिस सी की ओनली एंटीबॉडीज. वी डोंट हैव एंटीजन, है ना? हेपेटाइटिस सी की वन इज IgM, वन इज IgG, है ना? आई टोल्ड यू 3 पेयर्स एंड देयर रिलेवेंस. I told you the relevance. I told you S and antibody. Antigen antibody never occur together. This indicate infection. This indicate recovery. Recovery or vaccine. Say yes. Again, E never occur together. है ना? This indicate high infectivity. This indicate low infectivity. Say yes. And again, these never occur together. M indicate acute and G indicate chronic. Say yes. Question कैसे आएंगे इन पे? Let me show you question. ये question you will not get in micro. These question you will get in medicine. So I have covered the portion of the medicine here. Are you getting my point? See the question. देखो question देखो. I will show the graph. Wait. Some students are asking for the graph. Let me finish this topic. I will show you the graph. ठीक है? If you have the doubt, then you can ask. ठीक है? चलो. See. Don't look at the answer. Don't look the yellow portion. Don't look. Okay. I'm having 1, 2, 3, 4, 5. 5 patients in my clinic. This is the report of the first. This is the report of the second. This is the report of the third. This is the report of the fourth. And this is the report of the fifth. Read the report and interpret. You are a doctor, no? Imagine your neighbor is having hepatitis B virus infection, HBV virus. and he come to know that you are a doctor, you are a medical student, I mean. So he has done the serological markers and he's showing you a report. Doctor, can you please interpret my report? So how to interpret that? Do you know how to interpret that? Huh? Let me show you. See. First see S ki pair. S ki pair mein one of them is positive, one of them is negative. Then see E ki pair. One of them is positive, one of them is negative. And then see C. C की कौन सी antibody है? First S की pair में antigen is there, antibody is not there. Means person have the infection. मुझे तो समझ में आया कि person को infection तो है, but whether acute or chronic I don't know. Infection है ना? E की pair देख के, this is positive, this is negative. Antigen is positive, antibody is negative. Means person is of high infectivity. So person को infection भी है, the person is have the infection, the person is having high infectivity, and it's IgM. So say complete thing, पूरा word बोलो. Say, ma'am, it's acute hepatitis B virus infection of high infectivity. This is my diagnosis. This is how I interpret. How many of you got it? Yes, absolutely right. Absolutely right. Hana? Absolutely right. Arunan Shalam, Canal, absolutely right. And you get all these four options. You will get confused if you don't know the interpretation. You will get a preclinical scenario. There is a 57-year-old alcoholic man, diagnosed case of hepatitis B. He's not vaccinated and he's having high grade fever. He's having jaundice. He's having yellowishness of the sclera. A big clinical question. And they will give you the serological markers. Hepatitis S antigen is positive, antibody is negative. E ka antigen is positive, antibody is negative. And anti-HBC mein IgM is positive. What is your diagnosis? You will get these four options. Hannah, got my point. What should I repeat, Harsh? You have to be specific. What should I repeat? What should I repeat? I guess you want this interpretation again. So I guess you know the relevance. So let me tell you the interpretation. First see S का pair. Harsh, S का pair देखो. S के pair में antigen है कि antibody? हाँ? You people understand हिंदी? हिंदी में बोलूँ एक बार हिंदी में बोलती हूँ. S का pair देखो. हाँ? एंटीजन है कि एंटीबॉडी है? तो एंटीजन है, एंटीबॉडी नहीं है. इसका मतलब इन्फेक्शन है. इन्फेक्शन तो है, रिकवरी नहीं है. तो इन्फेक्शन है पक्की बात, रिकवरी नहीं है पक्की बात. चलो ई की पेयर देखो. ई के पेयर में भी एंटीजन है, एंटीबॉडी नहीं है ना? दोनों में से एक ही होगा. ओके, दोनों में से एक ही होगा. तो एंटीजन है, एंटीबॉडी नहीं है. ई की पेयर क्या बताता है हमें? ई की पेयर बताता है इन्फेक्टिविटी कितनी है. तो मुझे समझ में आया कि इन्फेक्शन है. यहां से मुझे पता चला इन्फेक्शन है ना? यहां से मुझे पता चला कि वो हाई इन्फेक्टिविटी का है, क्योंकि एंटीजन मतलब हाई इन्फेक्टिविटी, एंटीबॉडी मतलब लो इन्फेक्टिविटी. आई विल रिपीट इन इंग्लिश आल्सो. वेट जस्ट अ मिनट, है ना? ठीक है. और अब सी देख लो. सी मतलब आईजीएम. आईजीएम होता है एक्यूट. तो पूरी बात बोलो. कहो कि मैम एक्यूट इन्फेक्शन है. पहली बात तो एक्यूट है और इन्फेक्शन है और हाई इन्फेक्टिविटी का है. तो वही तो लिखा है, एक्यूट इन्फेक्शन ऑफ हाई इन्फेक्टिविटी. समझ में आया क्या? हर्ष, शिवम, कौन कौन पूछ रहा था? समझ में आई क्या बात? हाँ, समझ में आई क्या बात? से समथिंग. सो रमाया, इट इज फॉर यू. इन इंग्लिश आई एम रिपीटिंग. लिसन, चलो लिसन हियर. देखो, सो फर्स्ट यू लुक एट द एस का पेयर. First you look at the S का pair, then you look at the E का pair, and then you look the C का antibody. Okay जी? First on S का pair, antigen positive or antibody, one at a time. So antigen is positive, it indicates infection. There is no recovery. Antibody means recovery. So I'm sure my patient having the infection, not the recovery. So as soon as the patient is showing to me the report, I'm saying you are infected, you are not recovered. Number one. Number two, I'm looking at the E ka pair. Here also antigen is positive, antibody is negative. So it indicates the patient is having high infectivity. If antibody is positive, means low infectivity. So I'm patient the patient, you have the infection, you are not recovered, and you are highly infective. You are highly infective. Then I'm looking at C key antibody. And a C antibody, it's IgM. It means acute. So my final diagnosis is acute infection of high infectivity. Got it? Who was asking? Ramaya, you got it? Others, all the students got it. Not even a single student who didn't got it. Huh? In the same way, see the second report. Here also patient have the infection, but this time low infectivity. This time again acute. So acute infection of low infectivity. See the third report. Again infection, again high infectivity, but this time chronic. Again infection, this time low infectivity, but this time chronic. See the last report. In the last report, see this pair, the most important pair. S का antigen is negative, antibody is positive. Antibody indicates recovery. It's recovery. So patient is in recovery. Say yes. Okay. What you are asking, Lalit, can you please ask again? If I'm wrong, then please tell me when I can get it's chronic. What you are asking, Lalit? Can you please repeat? यस श्रीनिकेत ई की एंटीबॉडी मतलब लो इन्फेक्टिविटी, ई का एंटीजन मतलब हाई इन्फेक्टिविटी. What should I scroll? Okay, wait. I'm not getting it when I'm scrolling. Can you copy and put it again? Hmm. Is it IgM acute and IgG chronic indicates in report? Yes, it is indicated in the report. Is it IgM is acute and IgG is chronic indicated in the report? Yes, of course, of course, of course. Okay, chalo. Anyways, so let's move ahead. I guess everyone got it, है ना? So that was hepatitis B. It was a dangerous virus, not dangerous, but a little bit difficult. So you get questions in the medicine, many questions in the medicine. So let me come on the D now. Hepatitis D virus, है ना? Out of the five, I'm teaching you two in detail, B and D, and remaining we have seen in the table, है ना? We have seen the comparative table. So let's come on the D. Let's come on the D. You know what is D? What is D? Hepatitis D. D for Delta. It belongs to Delta Viridae family. You know that? Say yes. Say yes. I'm teaching you hepatitis D virus. Hepatitis D virus. Listen my point very carefully. Listen my point. D is Delta. वो तो ठीक है. D is defective. You will say, ma'am, what is a defect? What is a defect? It's a defective virus. It's like defective. D for defective. Never forget. D for defective. You say, ma'am, what is a defect? The virus is very shy. कुछ लोग होते हैं ना बड़े शर्मीले. It's very shy. है ना? The virus required a friend. Shy people always require a friend. कि तू भी चलना साथ. You also come with me. Then only I will go. I will not go alone. So if the virus is very shy, so he always require a friend. He always require a friend to cause hepatitis. He alone cannot cause hepatitis. No, hepatitis D alone cannot cause hepatitis. It always require a friend virus. It always require a friend. Who is the friend? Hepatitis B virus. So B ke bina D cannot do anything. D will cause hepatitis in presence of B. Got my point? So hepatitis D virus, it's a defective virus because it always require a friend. Friend virus is known as helper virus. It always require a helper virus to cause the hepatitis, to cause the infection, and that helper is B. Got it? I can't see your checks. Wait. Ah, okay, got it. Anna, chalo. So that is the thing. Now listen. There are two possibilities. Listen, everyone. There are two possibilities. They are in front of you. There are two possibilities. Imagine a healthy individual. Healthy the person is absolutely healthy. So D and B are entering together. D is saying to B, you also come with me. We will enter together and cause hepatitis in this person. So both of them are entering together and causing hepatitis in a healthy person. It is known as co-infection. Co matlab simultaneous. They are entering simultaneous. Say yes, got it? That is co-infection. I know. But in super infection, the person is not healthy. The person is a carrier of hepatitis B. See, compare. Here the person was healthy. Here the person is a carrier of hepatitis B. So person is asymptomatic. The person is asymptomatic because the person is carrier, not a case. The person is a carrier inside which hepatitis B virus is already present. Say yes, hepatitis B virus is already present inside this person. He is a carrier. And in it, hepatitis D is entering now. And now hepatitis D is entering. This is known as super infection. What does it known as? This one is known as super infection. Super infection. This one is co-infection. In co, the two are entering together. In super, the B is already present and now D is entering. You may be thinking, ma'am, how does it matter? Ultimately, B plus D are present in the human body. Does it matter? Is it co or super? Yes, it matters. Yes, it matters. Why it matters? Because acute से डर नहीं लगता, chronic से लगता है. So the percentage of acute and chronic is different. So let me tell you the percentage of acute and chronic. So if it is co-infection, means they are entering together, है ना? And if it is a super infection, है ना? They are entering one by one. So in co-infection, the person is healthy. The person is healthy, and B and D enter together. ठीक है? And in super infection, the person is a chronic carrier of hepatitis B. Hepatitis B. And D is entering super. कहते हैं ना super top up. So it is super. Super. Now see the percentage. What is acute here and what is chronic here? What is acute here? What is chronic here? है ना? You will say, ma'am, here 90% are acute, only 10 are chronic. So हमें acute से डर नहीं लगता, chronic से लगता है. Acute is not scary, chronic scary. So scary is only 10%. I know it's dangerous. I mean, I know here the percentage is reverse. Acute is 10, actually 20, and chronic is 80. If you have trouble in 2080, make it 1090. कोई फर्क नहीं पड़ता. Actually, it's 2080, but you can reverse, simply reverse 10 and 90. And just for the sake of understanding. So here chronicity is 10%, here chronicity is 90%. So you tell me which is having better prognosis, which is having poor prognosis out of the co-infection and super-infection. You tell me. So which is having better prognosis? You tell me. So co-infection have good prognosis and super-infection have bad prognosis. Because here chronicity is only 10% and here chronicity is 90%. That matters. That matters. Yes. Got my point? Yes. Yes, yes. So in superinfection, only D enters, but B is already present in the human body. No? Yes, yes. So this is co and superinfection. The same story is written in front of you. Read the question, tell me the answer. We are done with hepatitis. Read the question, tell me the answer. We are done with hepatitis. Yes. Hepatitis A is transmitted by. What is the root? A. Hepatitis A is transmitted by. I'm waiting for your answer. Yes. Hepatitis A is transmitted by, very good, it's fecal-oral route. So if I change the question to E, again the answer is same, fecal-oral route, है ना? But if I change the question to B or C or D, what is the answer now? BCD का answer क्या होगा? What is the answer for B or C or D? What is the answer of B or C or D? Yes. In that case, answer will become blood root. Blood root matlab parenteral. Most common is parenteral. Absolutely right. Which is not true about hepatitis B. Not true. Not true. Which is false. Is it a DNA virus? True or false? It is true. It can be transmitted from mother to child. Vertical. It is true. है ना वैसे तो ये भी false है. Two things are false. Fecal-oral route. It is false. Reverse transcriptase. It is false. Reverse transcriptase HIV में होता है. So B and D both are false. है ना B भी false है, you are right, लेकिन D भी false है. So B and D, actually D भी गलत है ना B and D both are false. So two answers are there. ठीक है. This question will not come in your exam, but I'm telling you. चलो next question. Interpret. You do the interpretation. HBs antigen is positive, है ना? IgM antibody is positive, and hepatitis E का antigen is positive. You tell me. You give the interpretation. The four options are in front of you. Tell me the answer is A or B or C or D. I'm waiting. What is the correct answer? Yes? So C, antigen is positive. So obviously the patient have infection, not the recovery. M matlab acute. So I will remove the two options of the chronic. Now I'm left with two options. Acute. I know. And hepatitis E, antigen also positive. It indicates infectivity. So acute with high infectivity, B is my correct answer. It is not acute or chronic. Say yes. Everyone got it? So these type of questions are very common in your exam. है ना? So you can interpret only if you know the relevance. Otherwise you cannot interpret. वरना देखते ही रह जाओगे मतलब क्या है इसका. Got it? You cannot learn the combinations. You should interpret on spot. Got it, everyone? First antibody to appear. First antibody. सोच के बोलना. Don't jump. Jump नहीं करना है. First antibody. सोच के बोलना. C. एंटी E, C और S. पहले ये डिसाइड करो. E, C, S में से है कौन सी? तुम ABCD का आंसर बताओ. What's the correct answer? Yes, most of the students are right. I'm happy. So first antibody is C, and C की IgM. IgM and IgG are the type of the C. IgM is not a type of E. IgG is not a type of E. है ना? समझ में आ रहा है? So C is the correct answer. Okay, C is the correct answer. Which last question? So this is the interpretation, Harsh. Na, I told you ना. अगर S है means the patient have the infection. You got the interpretation ना. M है मतलब it is acute. It is acute. So this one is acute. है ना? अगर G होता तो it was chronic. अगर M को मैं replace कर दूं G से, so it will be chronic. है ना? और E का antigen represent high infectivity. यहीं पे antibody अगर कर दूं, antigen को मैं सिर्फ antibody कर दूं, it will be low infectivity. So we can have various combinations. So if you know the relevance, you can do that. है ना? चलो. So we are done with DNA viruses. Congratulations. All six are done. Not only this, RNA में से भी 4 हो गए. है ना? कौन-कौन से 4? एक कैल्सी कर लिया. ठीक है. एक पिकोरना, one of the picorna, I mean hepatitis A virus, है ना? And one of the flavi, hepatitis C virus. तो I mean A, C, D, E. Hepatitis B virus यहाँ का है, लेकिन A, C, D, E चारों यहाँ के. D for delta. तो यहाँ के भी थोड़े-थोड़े ये वाले हो गए, है ना? Now let me teach you few more RNA viruses. जो important है वो देख लेंगे. So here I will teach you first picorna में polio. Picorna में I am teaching you 2 virus. One I already taught you hepatitis A virus. It is picorna. One more, I'm teaching you polio. So I will teach you polio from here. Okay. After that, I will teach you HIV, retro from here. I will teach you orthomyxo and paramyxo from here. And here it's not written, one is rhabdo. So I will teach you rhabdo also. And also rhabdo, retro, ortho, paramyxo, and so these 5 viruses are still pending. So can we start? The 5 more viruses we have to cover. After that, virology will be done. So today we are going to finish virology completely. And tomorrow, the last section, parasito, done. Puri parasito in one day. Parasito and myco also. Yes. Parasito and myco. Anna. So we will start with myco in the morning. It will take just 1 hour. Anna. If we start at 9, at 10 we will finish. 1 hour. Just 1 hour. And maximum 1 hour, 15 minutes if we do the questions also. So in 1 hour I will finish myco. Okay. So you will have a satisfaction that everything is finished except parasito, and then we will start parasito, and till end of the day we will continue it. And parasito will require nearly 5 hours, maybe 4 5 hours something. With breaks it will be till in the evening. ठीक है? So that will be the plan. तो पहले माइको कर देंगे and then parasito पे आएंगे. Tomorrow. ठीक है जी? And complete micro will be done from my side. Okay जी? Okay. Today maybe the 5 more viruses I have to teach, Deva. 5 more viruses, you say 15 minutes each. है ना? 15 into 5. ठीक है? Maybe another 1.5 hour. है ना? On an average another 1.5 hour. Maybe till 6:30 approx. ठीक है? चलो. Let's start picorna. So let's start poliovirus. In the picorna, let's start poliovirus. Can I start polio? Can I start polio? So let's start poliovirus. Chalo. So poliovirus, there are 3 types of poliovirus. Wild poliovirus 1, wild poliovirus 2, wild poliovirus 3. We are having 3 polioviruses. Okay. What is the difference between the 3? See the diagram of the poliovirus. Now I'm teaching you RNA viruses. This is RNA. Okay. Okay, can you see VP1, VP2, VP3, VP4? There are 4 proteins. Based on VP1, they are of 3 different types. VP1 is different in them. VP2, 3, 4 is same. So VP1 is viral protein 1. Viral protein 1 is different. Viral protein 2, 3, 4 is same. Okay, okay, okay. I was the professor. Anyways, I'm teaching all of you here, no? Anyways, chalo, listen. So based on the VP1, they are of 3 different types. ठीक है वाइल्ड poliovirus 1 2 3 out of them only one is remaining in the world. 2 and 3 already eradicated from the world. We don't have 2, we don't have 3. The 2 is eradicated long ago in 1999 and 3 is eradicated 2-3 years back only, very recently 2019, but 1 is still present in the environment and we are going to eradicate it very soon. Till now we have eradicated only 1 disease from the world, smallpox. And after smallpox, the next disease we are going to eradicate is polio. But till now, it is not eradicated. We are trying so. There are 3 polio viruses. We have eradicated 2 and 3. 1 is still pending, and we are trying to do so by giving the vaccination, pulse polio immunization, and all. Got it? Can we proceed? So this is the introduction. What is the mode of transmission? Polio virus spread by fecal-oral route. It is not a respiratory virus. It's spread by fecal-oral route. See the diagram. Everyone on the screen. See, polio occurs in children less than 5 years of age. Number 1. So he is a child. You can see the diagram, the child is there. The child is eating the food contaminated with poliovirus, eating the food or drinking water contaminated with poliovirus. So the virus is going inside. The poliovirus is going in the intestine. After going in intestine, the virus is going in the pair patches. Pair patches क्या होते हैं? What are pair patches? The pair patches are the lymphatics. After that, it is going in the mesenteric lymph nodes. है ना? It is going in the mesenteric lymph nodes, then it is going in the blood. The virus is going in the blood. So first in the intestine, then the lymphatics, then the blood. From the blood, polio virus go only in one organ. Only it is having affinity, tropicity only for one organ. That is spinal cord. It go in the spinal cord and destroy anterior horn cell. It destroy anterior horn cell of the spinal cord. Person have paralysis. Anterior horn cell are destroyed. Person have paralysis. है ना? Paralysis. The paralysis is flaccid. It's flaccid paralysis. How many of you got it? So there are 4 phases in the poliovirus. You can see the natural course of the poliovirus. First is alimentary phase when it is present in the intestine. Second is lymphatic phase when it is present in the lymphatics. है ना? Third is viremia phase when it is present in the blood. And finally, neural phase when it is present in the spinal cord. And it causes paralysis. Say yes. Say yes. So this is the complete course you can see. Anna. So how does it enter human body? Say via fecal-oral route. Via fecal-oral route it enters. So virus is ingested via fecal-oral route. After that, it goes to the intestine, then it goes to the Peyer's patches, it goes to the lymph node, it goes to the blood, causing viremia, right? And after that, it goes to the spinal cord. It destroys the anterior horn cell of the spinal cord and leading to paralysis, that is flaccid paralysis. It is leading to the paralysis known as flaccid paralysis. That is the mode of transmission. Take care, read the question and tell me the answer quick. Then I will tell you ahead the polio and all. Read the question. What is the most common mode of transmission in poliovirus? Yes, most common mode of transmission. So of course the answer is fecal-oral. है ना? Fecal-oral route. ठीक है? Now, if a child have the polio, if the child have poliovirus exposed, है ना? The virus enters inside the child, poliovirus, and enters inside a child. So there are four possibility. One of it will occur. There is one of the four possibility. There is one of the four possibility. What are the one of the four possibility? What is one of the four possibility? 95% chances is that the person will have subclinical infection. Subclinical infection. 95% chances that the child will not have any symptom. Maybe बचपन में हम सबको हुआ. Maybe in our childhood we all have the polio. 95% is a big percent. The child will not have any symptom treated of its own. No treatment required. Child don't know even I have the polio. And a 95% chance. Only if you check the blood of the child, you will get the antibodies. Otherwise, child don't have anything. है ना? 4 to 8% chance child will have minor illness. Minor मतलब abortive. Abortive मतलब abortion. Minor illness. है ना? It is a self-limiting minor illness. है ना? The child will have a fever, a little bit of fever and flu-like symptom, nothing else. No treatment required. Antibodies are rest, automatically treated, self-treated. No treatment required. 1% chance the child will have meningitis. You know meningitis, the neck stiffness. So whenever the child will bend, the child will have neck stiffness. है ना? The child will have neck stiffness, meningitis. But again, it's treatable. It's treated. Treatment is given and it's treated. Less than 1% chance the child have the polio. So whenever we think about the polio, we always think about this. है ना? Polio word आते से हमें ये imagine होता है. But this जो imagine हो रहा है ना, it is less than 1. It is less than 1. 99% don't have the paralysis. है ना? Maybe हम सबको polio हुआ है बचपन में. We are coming in the 99% category, not in this less than 1% category. Less than 1%, you can say the unlucky one, they have the paralysis. You tell me the percentage. So whenever a polio virus enter in human body, in a child, so there are four possibilities. 95% chance the child is asymptomatic. It is known as subclinical infection. Say yes. 4 to 8% chance the child will have minor infection known as abortive infection. Say yes. Got it? So 4 to 8% chance. The next 1% chance the child will have meningitis. Okay, G. And less than 1% chance the child have paralysis. Take care. So please learn these percentage. Okay. Now let me tell you the paralytic. This less than 1 wala. I will explain you this in detail. Less than 1. Although they are less than 1, but they are the very unlucky people. There is no treatment for polio. So paralytic polio. It is only less than 1%. Less than. So it can occur in less than 1% of the people who are infected. The paralysis is flaccid. It is known as asymmetric flaccid paralysis, AFP. What is the full form of AFP? Asymmetric flaccid paralysis. Got it? That is paralytic polio. Let's describe paralysis. Listen, the paralysis is descending in nature. What do you mean by descending? It starts at the hip and gradually descend down, descend down, descend down. It starts at the hip and gradually descend down. That's why descending. It's asymmetric. Asymmetric means proximal muscles are involved more than distal. So the thigh muscles are involved more than the leg. The thigh muscles are involved more than leg. So that is asymmetric. It is not all the muscles are involved. The thigh are involved more than the leg. Got my point? Got my point? The next is DTR. The DTR is diminished. Okay, there is no sensory loss. The child is having the paralysis now. If you touch the child here on the leg, if you touch the child, heat, cold, everything senses are there. है ना? चिमटी खोलो, pinch करो, सब पता चलेगा. Sensation is normal. Motor loss is there. Sensory loss is not there. Motor loss is there. है ना? The limb is not used, so limb get atrophied. Can you see this limb? It's very thin. It's very thin. It get atrophied. It get atrophied. I know. So that is the features. Can we revise the features quickly? You say, ma'am, paralysis is descending. It start at the hip and gradually descend down. It's descending. Number one. Number two, say, ma'am, it's asymmetric. The hip muscle are involved more than the leg muscle. It's asymmetric in nature. and atrophy is there. There is no sensory loss. DTR is diminished. It is known as asymmetric flaccid paralysis, AFP. How many of you got it? How many of you got it? Read the question. Tell me the answer. Quick. All are true about the polio except, I'm saying except. What is not true for the polio? Tell that. So very correct. So you are saying 90% is paralytic. No way. Less than 1% is paralytic. So this statement is absolutely false. The remaining three category are non-paralytic. You can say 99% is non-paralytic. Yes. It is flaccid. Yes. Aseptic meningitis occurs. Yes. But never. It is less than 1% is paralytic, not 90%. So correct answer among them is D because they are asking the except. So correct answer is D. Right? There is no treatment for polio. There is no treatment. Once polio occur, it will be lifetime. No treatment. You can give the physiotherapy. It's a tertiary rehabilitation. I know that's it. But there is no treatment. The child will have paralysis throughout the life. But prevention is possible. We have the vaccine. We don't have treatment. We have the vaccine. We have the vaccine. So all the children who are less than 5 years of age should get the vaccine of the polio. Must get the vaccine of the polio. Two types of vaccines are available. Diagrams are opposite लग गए गलती से by mistake. Oral 2 बूंद जिंदगी की. 2 drops are given orally. The punchline is do boon zindagi ki. You may have heard that. So that is 2 drops given orally, oral route. And second is injectable. The injection is given. Injectable. Images are opposite by mistake. So this is injectable. है ना? So oral polio vaccine is known as OPV. Oral polio vaccine. And injectable is known as IPV. Injectable polio vaccine. Oral polio vaccine. Injectable polio vaccine. OPV and IPV. Got my point? Say yes. Have you read this in PSM? Shall I skip here or you want me to tell you? You want me to tell? I will tell. It's okay. Huh? You want me to tell the differences between OPV and IPV? Which is better? What are the pros and cons? What are the advantages and disadvantages of each of them? Okay. We will start. Listen. OPV and IPV. Say the full form. Say the full form. OPV is oral polio vaccine. The route is oral, obviously. IPV is injectable polio vaccine. The route is injectable. Injectable. Intramuscular, intradermal. Both will work. But it's injectable. Okay? Listen my point. Oral is live. We are giving live virus. That is attenuated. Live attenuated virus. And here it is killed. In injection, the virus is killed. The virus is same. But here it is live attenuated. Here it is killed. Learn the name of the scientist who discovered it. The OPV is discovered by Sabin. That's why known as Sabin vaccine. And this one is discovered by Salk. That's why known as Salk. How you learn that? How I learned? Let me tell you. Salk. Salk ka spelling kya hai? SALK. LK. KL. KL. So Salk is killed. I learned like that. I never forget. Salk is killed. Salk mein LK. LK se I remember KL. KL matlab killed. So killed wala Salk hai. Or killed wala to IPV hai. I know that. So this is how I correlate. You got my point? You got my point? You tell me which is better. This or this, and the reason why. Which is better? Which is better? So let me tell you, both are better. I mean, there are some advantages here, which are disadvantages here. There are some advantages here, which are disadvantages here. So it's vice versa. So let me explain you both. In one diagram, I will explain you both. Everyone on the screen. Everyone on the screen. Fatafat in 1 minute, I will explain. Do boon zindagi ke. Let's give OPV to this child. OPV. What does OPV contain? Live virus, which is attenuated. So let me tell you the advantages, 5 advantages. Listen, oral route is very easy. So you see, ma'am, easy to give. Child is, I'm sorry. Child is very happy taking the oral drug. If you give injection, the child will cry. Just a second. I will have cough otherwise. That's why I'm taking constantly some lubricant so I can have. Anyways, chalo. So it's very easy to administer to the child. If you give it to the child is easy. It is easy for the parents also, easy for the child. So easy to administer. Number 1, first advantage. Easy to manufacture also. Easy to administer also. Easy to manufacture also. Okay? After giving, where does it go? It will go inside like this, and it is excreted in feces. Some of it is excreted in feces. It will go in the lymphatics. Some of it will go in the blood. But it cannot go in the spinal cord. Of course, because it is attenuated. Attenuated means weakened. Attenuated means weakened. It cannot go in the spinal cord. Got my point? It cannot go in the spinal cord. Otherwise, it will cause paralysis. It cannot go. So it will remain in the blood. Got my point? So listen now. Listen what I'm telling you. In the intestine, body will form IgA antibodies. Have I taught you antibodies on day one? There are 5 types of gamma, IgG, IgA, IgM, IgD, Ig. बताया था क्या है ना? A is present in the secretion है ना? तो intestinal secretion में A बनेगा. A is formed here and remaining all पांचों के पांचों यहाँ भी बनेंगे है ना? I mean to say it is giving double immunity. Local immunity in the intestine and systemic immunity. Say double immunity. Local immunity in the intestine and systemic immunity in the blood. You will see, ma'am, it is also very good. So easy to give, easy to administer, easy to manufacture, and let me write, it's cheap also. It's very cheap also. Yes, this one is cheap. Hannah, it's very cheap. So it is also good. It's very cheap. Hannah, double immunity is also given. It is also good. One more thing. Hannah, listen. It is excreted in feces. Hannah, have I told you it is going in intestine? So ultimately it is excreted in the feces. Now, listen. Just suppose I'm doing a camp. I'm doing a camp in a village. There are 100 children in the village. So I asked, or there are 100 children who are less than 5 years. So I asked all the 100 should come in my camp and I will give OPV to all of them. But instead of doing so much of awareness, so much of publicity, only 40 children came for the vaccination. The 60 do not came. It's okay. It's okay. So you give the 40. You give OPV to the 40. No worries, you give. What does these 40 will do? These 40 you have given do boon zindagi ke to all these 40. I know. So they will excrete the virus in their feces. I know that day or next day of the camp or that day only. I know they will excrete the virus. The feces will contaminate the food and water. I know. And the contaminated food and water is taken by another child. And in the villages, due to overcrowding and what sometimes poor hygienic condition, the contamination is very common. This is the only place where we want the contamination. वहाँ पे unvaccinated child also get the vaccination via contaminated food and water. The vaccinated child, this child is vaccinated. The vaccinated child is excreting the virus in the feces. The feces will contaminate the food and water, and the contaminated food and water will be taken by non-vaccinated child. So non-vaccinated child तक भी हमने vaccine पहुंचा दी बिना हमारे जाए. How many of you got it? How many of you got it? So you can write the advantage. Non-vaccinated child will also get the vaccine. So write down what you can write here. I mean, non-vaccinated child will also get the vaccine. It is useful in herd immunity. इसी के reason से. It is useful for epidemic. इसी reason से. Useful in herd, useful in epidemic. It is non-vaccinated child can also get the vaccine. There is a chance. I mean, so these are the advantages. Hannah, these are the advantages. So can you enumerate the advantages? Let me highlight the 5 or 6 advantages. See, number 1, easy to give, easy to administer. Number 2, easy to manufacture. Number 3, it's cheap. Number 4, double immunity. Hannah. Number 5, non-vaccinated can also get the vaccine. Number 6, provide herd immunity. Number 7, useful in epidemic. आप बोले मैम सब बढ़िया ही बढ़िया है. Problem क्या है? What is the problem? There are only 2 problems. Only 2 disadvantages. Number 1 disadvantage, storage is very difficult. For storage, the temperature required, see the temperature, -20 to -40. What is the temperature of your fridge? Fridge में होता है नीचे होता है 2 to 4, ऊपर होता है 0, है ना 0. हमें चाहिए -20 to -40. So we require deep freeze. Deep freezer. Normal fridge से काम नहीं चलेगा. So it's very expensive to store, or I mean not expensive, it's difficult to store. And it's very difficult. If this temperature is not provided, now it is ineffective. It will become ineffective. Got my point? Storage is difficult. Number 1. Number 2, I told you it is live but attenuated. So after blood, it cannot enter head. But sometimes attenuation is not very good. Attenuation is not very good, and it can enter in the spinal cord and cause the paralysis. Such a paralysis is known as vaccine-associated paralytic polio, VAPP. What is it known as? VAPP, वैक्सीन एसोसिएटेड पैरालिटिक पोलियो, VAPP. सोचो वैक्सीन की वजह से पोलियो हो गया. The polio occur because of vaccine. You are getting my point? We give vaccine to prevent polio. Imagine उन parents की क्या हालत होगी. Imagine the child. वो parents तो इतने अच्छे हैं. They are very aware. They are giving the vaccine to their child on time. But because of the vaccine, their child has the polio. It is known as VAPP. So although the chance is very rare, 1 in 1 million, the chance is 1 in million, but वो million में से भी कोई एक child है, किसी का तो child है ना जिसको हुआ. In India till now 4 cases are there. VAPP, very dangerous, है ना. So these are the 2 disadvantages. Say yes. How many of you got it? So the 2 disadvantages, storage and VAPP, the chance of VAPP. So 6 advantages, 2 disadvantages. I will do the reverse. in IPV. Can I flip? Can I flip? Say yes. So let's flip. Let's flip. Let me tell you IPV. IPV is given directly in the blood. It is not given orally. It is given directly in the blood. है ना? It is given directly in the blood. This is IPV. है ना? So the first disadvantage, difficult to give. है ना? Child will cry. Injections are not good. है ना? Number one. Difficult to manufacture also. Number two. It is expensive also. Number three. ठीक है? These all are disadvantage. Number four, it will give only one immunity, the systemic immunity. It will not give local immunity because it is not going in intestine. No local immunity, only systemic immunity. है ना? It is not excreted in feces. It is not going in intestine. So no, not useful in herd immunity. Okay, not useful in epidemics. Okay, and non-vaccinated child cannot get vaccinated. जिसको दे रहे हो सिर्फ उसको मिलेगा. It is not transmitted to the non-vaccinated child. ठीक है? So all the advantages convert into disadvantages. But there are two advantages. जो वहाँ पे disadvantage थे, they become advantage here. Number one, storage is good. है ना? For storage, we require 2 to 8 degree. And not minus 20 to minus 40. It requires only 2 to 8, not minus 20 to minus 40. So storage is not difficult. Number two, the virus is killed. There is no chance of VAPP. No chance of VAPP. So these are the two advantages. So advantages are only two here. Storage is not stringent and VAPP ka no chance. Remaining all are disadvantage. How many of you got it? So that is the thing. You can read it by your own. You want me to write or you got it? So there are two types of vaccine. You can write OPV, IPV. Anna, OPV, IPV. Tell me which one is live. This one is live. This one is killed. Tell me the route. This one is oral. This one is injectable. Tell me the scientist. This one is Sabin. This one is Salk. LKKL. Learn like that. Anna. Now you tell me, manufacture किसका easy है? एडमिनिस्ट्रेशन किसका इजी है? एडमिनिस्ट्रेशन, मैन्युफैक्चर, कॉस्टिंग बताओ दोनों की मुझे, है ना? इम्यूनिटी बताओ, सिंगल और डबल, लोकल और सिस्टमिक, है ना? इम्यूनिटी. उसके बाद हर्ड इम्यूनिटी बताओ, उसके बाद एपिडेमिक बताओ, है ना? नॉन वैक्सीनेटेड चिल्ड्रन वाला बताओ, स्टोरेज बताओ और VAPP बताओ. So all the points yes, you can write. Okay, I will write it very quickly. Manufacture, this one is easy. Administration also, this one is easy. This one is difficult. है ना? Costing also, this one is cheap. This one is expensive. है ना? Immunity, it provides double. Systemic and local. This one provide only systemic, no local. है ना? Herd immunity, epidemic. Here it's good. It's useful. I mean, herd immunity and epidemic. It's not useful. है ना? Non-vaccinated children also get vaccine. Here there is no chance. But the two things. Storage is stringent here, and storage is not stringent here. है ना? नंबर टू, VAPP का चांस है यहाँ कोई चांस नहीं है. How many of you got it? How many of you got it? So this is the thing. You will get repetition in the PSM also. Okay. Have I taught you only PSM to this batch? I'm asking. PSM is taught by me only in this batch or someone else? I'm not remembering because that's why. So I have taught you PSM. Then I have taught you this in PSM also. So again and again, I'm telling you the PSM faculty will teach you. I don't remember that I'm only the PSM faculty. Anyways, chalo. I have taught you the same slides in PSM also. So you can revise there also. So can we proceed ahead? Okay, chalo. So let's proceed. So we are done with polio. We are done with one of the virus, polio. Let's come on the next rhabdo. Can I start rhabdo? Can we start rhabdo? Huh? We have to study rhabdo. After rhabdo, HIV, retro, and after that, orthomyxo, paramyxo together. So 15, 15, 15, maybe 45 minutes, maybe 1 hour. है ना? Prophylactically 1 hour. Can we take a small break? Again 10 minutes, 5 minutes or 10 minutes. After that 1 hour, the last 1 hour. ठीक है for today. Can we take? Huh? Okay. So it's 5:11. So 5:10, 5:20, 5:25. So can we start at 5:25? Anna? Okay, ji? Anna? Okay. So let's come at 5:25. Okay. So I'm not ending the session. I will continue in this only, or I will ask the team if they want to break because sessions are very long now. So recordings are not available or some problem. So they break it and again create new link. Let me ask. Otherwise, same link. We will continue. Okay, ji? Thank you.